So good morning, everybody, and welcome to the next in this series of highlighting organic chemistry across Europe. And we're delighted this morning on 8th of November 2024 to highlight organic chemistry in Catalonia. And this has been organised by the Chemical Society of Catalonia. And it's a great pleasure to introduce an old friend, but also a member of the EuChemS division, Professor Anton Vidal, who will give some introductory remarks about the importance of organic chemistry in Catalonia and introduce each of the speakers. So good luck to everybody, and I'm looking forward to this symposium. Thank you very much.
Okay, good morning, everyone. My name is Anton Vidal, and I serve as the representative of the Catalan Chemical Society in the Division of Organic Chemistry of EuChemS. It's been my privilege, alongside the boards of our society and the Division of Organic Chemistry of EuChemS, to contribute to the organization of this event. Let me begin by expressing my gratitude to all of you participating in this online symposium, which aims to showcase the research in organic chemistry taking place in Catalonia. As the first speaker, my role is to provide a brief introduction to the Chemical Society of Catalonia and highlight research institutions in organic chemistry within Catalonia. I'll try to keep my remarks concise so that we can swiftly turn our attention to the true stars of today's events, which are the speakers. So here are some historical milestones of our society. The Catalan Society of Chemistry has its roots in the early 30s, when scientists from our country established the Catalan Society of Physical, Chemical, and Mathematical Sciences. This organization functioned at that time as a subsidiary of the Institute of Catalan Studies, l'Institut d'Estudis Catalans. In 1986, the Catalan Society of Chemistry became an independent entity while maintaining its affiliation with the Institut d'Estudis Catalans, which continues to provide vital patrimonial, legal, and logistical support. The Catalan Society of Chemistry was formally accepted as a full national society member of EuChemS in 2007. And the last milestone is that the youth section of our society was created in 2022. And the main objective of this section is to bring together young chemists eager to propose and organize scientific activities in the field of chemistry, making full use of the various current technology platforms. Today, the Catalan Society of Chemistry boasts or has approximately 900 affiliated members. A notable strength of our society lies in the diverse membership, encompassing professionals from academia, industry, and high schools. The communication channels of our society include a monthly electron bulletin, our website, and the platform formerly known as Twitter. So the map highlights the geographic area of influence of the Catalan Society of Chemistry, corresponding to the current political boundaries of Catalonia within Spain. Instead of marking major cities, the map highlights the location of universities offering chemistry programs at the undergraduates, masters, and PhD levels: University of Girona, Black Dot; University Autònoma of Barcelona, located in Cerdanyola del Vallès, Green Dot; Universitat Ramon Llull, this is a private university in Barcelona; Red Dot, University of Barcelona, which is the largest, and this is the light blue dot; and Universitat Rovira i Virgili in Tarragona, which is the purple dot. So in this extended map, I have highlighted other areas in Spain, France, or Italy where variants of the Catalan language are spoken, while currently we have a few affiliated members of these areas, fewer, I should admit, that we would like. We are actively working to increase membership in these areas. Notably, 2 members of our board come from the regions marked in gray. I have highlighted a few of the many scientific events organized by our society. The first is the meeting of young researchers, which bring together PhD students and postdoctoral researchers conducting their work in academic settings. This event is held biannually, with the most recent edition having taken place earlier this year in Tarragona. Yeah? An initiative introduced by the current board includes biannual meetings focused on theoretical and computational chemistry, inorganic and organometallic and organic chemistry. Yeah, and the next editions of these meetings are scheduled for next year, sorry. Additionally, the Catalan Society organizes or participates in various events such as the commemoration of the Nobel Prize in Chemistry every year and general chemical debates. For the sake of brevity, descriptions of these events are not mentioned. The Catalan Society actively promotes the publication of several scientific journals, books, and other educational materials. While it's not possible to mention them all, I have selected three that, in my opinion, are the most representative. The Journal of the Catalan Chemical Society, which aims at serving as a communication channel among the society, and it aims to disseminate and promote scientific research in any branch of chemistry. The second one is Educació Química, EduQ, is a journal on chemistry education published by the Catalan Society, and it features original articles at all educational levels, particularly focusing on secondary education. Yeah, and then the third one is the publication Classics of Chemistry, and the aim of this collection is to introduce chemists from Catalan speaking territories to key texts that have revolutionized the field of chemistry and are essential for understanding its development. Yeah, the picture on this book does not need any more explanation for the organic chemistry community. Yeah, the Catalan Society of Chemistry is active as well in recognizing excellence through a variety of awards, which I'll now present. The first group, Scientific Excellence and so on, focuses on academic achievements and is tied to research activities. The second group recognizes educational excellence and innovation, encompassing both university and high school. Additionally, the Society actively engages in outreach initiatives aimed at both high schools and the general public. The Catalan Chemical Society also coordinates the recognition of historical chemical landmarks in Catalonia, further highlighting our rich scientific heritage. And regarding awards for high schools, the Catalan Chemical Society honors...
Perfecte, però veus perquè puc compartir?
Pues no et senten.
Honors... perdó, Òscar, us esteu sentint. Rewarding awards on recognition for high school students, the Catalan Chemical Society honors outstanding research projects and videos submitted by high school students. Yeah? As for teaching and research in chemistry, 12 universities in Catalonia has 12 universities. 5 of them offer comprehensive chemistry programs at the bachelor, master, and PhD levels. The different universities are listed in the screen. Yeah. As well, the Spanish Research Council has a significant presence in Catalonia with several high-level research institutes. Yeah, depending on the source, the number of these institutes is reported as 46. However, this figure can be misleading, as the CSIC, National Research Council, is often counted as a single entity despite managing several research centers. Yeah? The Catalan Institution for Research and Advanced Studies, ICREA, has also played a pivotal role in advancing research activities. And although ICREA does not manage its own research institutes, it employs today around 300 research professors who conduct their work within university departments and various research institutes. Yeah? The second ICREA initiative is the ICREA Academia program, and a heavy teaching load of academics often limits the time university professors can dedicate to research. The ICREA Academia program helps alleviate this burden by reducing the teaching responsibilities of awardees of this program, allowing them to focus more on their research activities. I'm hearing someone. Please mute the microphones. Yeah. In total, around 13,000 individuals are engaged in frontier research across university departments and research institutes in Catalonia. Unfortunately, I do not have specific data of how many of these professionals are directly involved in the field of chemistry. Yeah. The last slides refer to today's event, highlighting organic chemistry in Catalonia. And today's program features 7 lectures delivered by outstanding speakers conducting research across 4 university departments and 3 research institutes. The program seeks to showcase research in organic chemistry, not only as a stand-alone discipline, but also as a field deeply connected with other disciplines such as biochemistry, biology, nanotechnology, materials, and so on. The research presented will underscore the essential role of organic chemistry in advancing knowledge and innovations. Yeah? The institutions where the speakers conduct their research are listed in the slide, following their order of presentations. And last but not least, we extend the organizers, extend the apologies to the many research groups whose outstanding work could not be featured today due simply to time constraints. And it's important to acknowledge that numerous other groups from various chemistry departments and research institutes also deserve recognition and would deserve having a time slot today. And with that, let's dive into the program and let's get started. Now I stop share, yeah, so that the first speaker of today's program is Daniel Maspok, who performs his research activities at the Catalan Institute of Neuroscience and Nanotechnology in the influence area of the university, in the campus, located in the campus of the Universitat Autònoma de Barcelona, and he's a research professor from ICREA. The title of his talk, as you can see, is Clipping of Chemistry, Synthesis by bond cleavage. Daniel, the floor is yours.
Okay, thank you very much, Anton, and good morning to everybody. And first of all, what I would like is to thank Anton for inviting me to this highlight organic chemistry in Catalonia. I mean, it's a really honor. I'm usually invited in organic chemistry conferences, so I think it's my first time doing it in an organic chemistry, so thank you very much. So basically my lecture today, what I want to try to explain is our first steps towards the development of a new synthetic strategy that we have called clip of chemistry. So overall, we define clip of chemistry as a synthesis of molecules and materials with well-defined structures and compositions through the orthogonal bond cleavage within molecular structures. So from this definition, I think it's quite clear that if we want to construct fragments of molecules or new materials using a cleavage reaction, it's necessary to start within a molecular material that contains repetitions of these fragments. And for this reason, like 8 years ago, we decided that reticular materials will be the ideal precursors to start developing this new synthetic strategy. And since now reticular materials will be very important in this presentation, I want just to dedicate 3-4 slides trying to introduce what are these type of materials. So basically, reticular materials are crystalline frameworks that are constructed through the rational linkage of molecular building blocks together by strong bonds. And by strong bonds here, I mean coordination and covalent bonds. There are two main types of families of reticular materials. So the first ones are the ones in which we are linking an organic molecule and a metallic component that can be a metal ion or a cluster. And when you combine these two building blocks together by coordination chemistry, you can construct two or three-dimensional porous crystalline structures that are well known as metal organic frameworks, or you can construct finite structures that are known as metal organic cages. Now, if you replace the inorganic component by another organic molecule, and you combine these two organic molecules using dynamic covalent chemistry, you are constructing the same type of structures, but made of purely organic components. These structures are known as covalent organic frameworks or covalent organic cages. All these family of materials, they share many different important properties. For example, they are quite thermally stable. They can be stable up to 300 to 500 Celsius degrees. They can be also chemically stable. They can be stable in water, in many organic solvents, even under acid or basic conditions. But I think the most popular property of these materials is that they are highly porous. They can exhibit surface areas in between 1,000 to 7,000 meters square per gram, which means that 1 gram of this material, they have the same surface area of a professional soccer field in order to start absorbing, storing, releasing different type of molecules that can range from gases to drugs, for example. But these three properties were not the ones that attract our attention for our purpose, trying to demonstrate the synthetic approach. What attracted us, the attention, was that they have a very wide compositional and structural diversity. What does it mean? Basically, it means that through inorganic chemistry, we have a wide variety of metal ions as well as clusters that we can use. They can have different compositions. They can be made from copper, iron, many other. They can have many different connectivities and as well many different geometries. Equally, organic chemistry offers us a lot of different possibilities of designing and synthesizing organic molecules that can be used for this purpose or to make these materials. They can have different functional groups like carboxylic acid groups, phosphonic groups. They can have different geometries and as well many different connectivities. Now, it's like playing with a Lego in a child. You just need to start combining all these building blocks together. And if you do this, you can make a large variety of different materials and structures that will have many different structures. However, what attracted us more is that the fact that when you are linking these very basic building blocks, in fact, what we are constructing inside these structures is repetitive units or fragments that can be very important by themselves. And we can find inside these structures many different types of fragments that can be very simple ones like new molecular clusters or secondary building blocks, or can be much more complex like cages, microcycles, nanowires, polymers, layers, many of them. Let's focus a bit more in this particular aspect and concept. So let's imagine now that we have a zirconium metal ion, very simple one, and terephthalic acid. If we combine these two elements, most likely we are going to construct this three-dimensional structure. And when this zirconium and this terephthalate link together, what we are making is this new 12-connected hexanuclear zirconium cluster that they have very interesting catalytic properties by themselves. But if we now change the synthetic conditions or we slightly change the linker, we can spontaneously make many other different clusters that are not existing or that cannot be synthesized from a bottom-up point of view. We can keep the same connectivity by changing the geometry, or we can even decrease the connectivity and the geometry. And this is the simple building blocks. There are many other structures in which we can find metal organic and purely organic cages, like this octahedral cage. But if we check the literature, there are many other different structures in which we can find many different types of cages that are made spontaneously by linking these two simple building blocks. We can find very simple ones like this tetrahedral cage, or very complex one like the endecahedral cage. And we can even increase the connectivity or the dimensional, and we can start finding very interesting one-dimension polymers or even metal organic wires, two-dimension organic or metal organic sheets, or even we can find three-dimensional structures that are hidden inside other three-dimensional structures. And it was at this moment that we saw that all these repetitive units can, in fact, have very interesting properties and function by their own. So they really have the potential to serve as a new type of molecules and materials if we are able to isolate, to release and isolate from all these reticular materials. And it was at that moment when we figured out this that the clip of chemistry concept arose in our laboratory. To try to develop a new chemistry in order to release and isolate all these repetitive fragments from these reticular materials. How we visualize this clip of chemistry concept. So now imagine that we have this, imagine a purely organic framework, and we want to isolate this covalent organic cage, okay? And we can simplify this organic cage with this Lego bridge that is formed by this trigonal red brick together with these small blue pieces, okay? So this is our target molecule. And now imagine that we have the simple molecular building blocks, which is, for example, this trigonal red Lego brick and this linear blue one. When we link these two building blocks together, most likely we are going to create this two-dimensional honeycomb structure. And when we create this structure, you can clearly see that, in fact, we are making repetitions of our target cage. How we propose to release and isolate this cage. So basically what we propose is to encode one of the organic linkers with a cleavage site. If we do this, and now we use this new linker, which will be very similar to the other one, but containing a cleavable bond, and we combine again these very simple building blocks, we are going to construct again this honeycomb layer, but now it will introduce these cleavage sites. In the second step, what we propose is to use a cleavage reaction in order to break or to cleave all these linkers in such a way that we will release this repetitive unit and isolate the cage. So it's quite simple that our strategy has two very important steps. First one is to encode with cleavage sites, and the other one is to break these bonds using an organic reaction. And for this reason, at that moment, it was very important to decide which will be our cleavage reaction, with our initial cleavage reaction and our initial cleavage bonds. And for this, we decided as a first option that a good reaction will be ozonolysis. Why we decided that ozonolysis will be a good choice to start developing this approach? Basically because it offers three main advantages. The first one is that it's a quite selective reaction. It means that when we have these olefinic bonds and we expose to ozone, we can oxidative cleave them, forming this 1 to 4 trioxylene ring that it's quite unstable, and it breaks, forming an aldehyde and a carboxylic acid groups. Second reason is because it's a gas-solid reaction. And if you remember, we are using porous materials. So we thought that using a gas as a reactant will be much easier to diffuse along the channels and reaching the many different olefinic bonds that will be placed in the structure. And finally, we can somehow control the output of the reaction. That it means that using reductive working up or oxidative working up conditions, we can control the cleavage of these olefinic bonds only into aldehyde groups or carboxylic acid groups. And these are very important conditions if at the end we want to synthesize a molecule or a material with a well-defined structure and especially composition. So now it seems that we have everything. We have now the cleavage site, we have the cleavage reaction. So we are at the moment in which we can start now taking all the many hundreds and thousands of different reticular materials and use them as precursors to start synthesizing new molecules and materials in a kind of top-down approach control at the molecular level. And for this, I'm going to try now to explain some examples. The first one will be more based on metal-organic structures, and at the end will be purely organic ones. Just trying to demonstrate that indeed it's possible to start using this strategy, this new strategy to make new molecules. And when we start this, it was around 8 years ago, and we start having the first results, it looked logical to start proving the efficacy of this strategy using the simple model as possible. And for this, we decided that a very simple model, and the first one we try, will be this cage. Okay, so this is a cage, very simple molecule. Here is defined. It's formed by two rhodium paddle wheels, okay, one on the bottom, one on the top, and they are linked by these 4 organic linkers, and each one of these linkers contains 2 olefinic bonds. What it means that if we want to be successful with our approach, we need to simultaneously break 8 different olefinic bonds. And if we do this, each one of these linkers will be cleaved into this 3,4-methyl benzoic acid that will be keep coordinated to the rhodium ones, and we will release these isophthaldehyde molecules. And at the end, the product that we expect will be this rhodium paddle wheel cluster with these 4 phenyl rings that will be functionalized with aldehyde group at the meta position. Okay. This will happen if we use obviously reductive conditions forming aldehyde groups. Now, so basically this is what we did. First, we synthesized this cage. This is very simple. And we tried hundreds of different conditions. Here, I'm just going to explain the optimized ones that we found out that when we dissolve this cage in a dimethylacetamide and dimethyl sulfide mixture, adding a bit of reducing agent, we can start ozonizing. How we ozonize? It's a very simple concept. Basically, we have ozonator that produces ozone. It passes through the calcium chloride path in order to dry as much as possible the ozone, and then it flows to the place where we have the reticular material that can be in solution, bubbling ozone, or in solid state, and at the end we have a bath indicator in order to know when the reaction is finished. So this is the model. This is how it looked like in the lab, where we have all the ozonators, the ozone flowing to the gas system, where we can mix it with different gases, and then it directly flows to a dispersion or a solution when we bubble ozone, or here that we have in solid state. Of course, we have the detectors in order to know which is the concentration, more or less concentration of ozone that we are using in each one of the reactions. So basically, this is what we did here. We place our cage here, and we start bubbling ozone for 6 minutes. At the end, you obtain a very clear solution. You can precipitate the solid using chlorhydric acid, take the solid, and you can easily recrystallize in a mixture of water acetone, obtaining these beautiful crystals in a 95% yield. Now, if you solve the crystal structure, you can really see the formation of this rhodium paddle wheel. Now, with these 4 phenyl rings functionalized with an aldehyde at the meta position. And you can now start from this very simple concept or this simple example, you can now start extending this type of chemistry in order to make many different types of complexes. Like, for example, this cluster that contains 2 aldehyde at the meta position, but 2 other at the para position, you can start making crown type complexes or even new type of microcycles only and basically using more or less the same precursor by changing slightly the conditions or the additions of these cleavable bonds inside the cage. You can also apply now this chemistry to 3-dimensional framework. So imagine that we have a 3-dimensional framework, we have a crystal, and now we are able to isolate the molecular clusters that are forming this framework. This is one example in which we, just by basically mixing this scandium salt and this dicarboxylic acid, you form a 3-dimensional structure in which we spontaneously form trimeric clusters that have not been synthesized from a bottom-up point of view, but that can be easily formed when we mix these 2 simple molecules. And we encode this, as you see, each one of the linkers with olefinic bonds. After making this, you can now start applying ozone and release and isolate the corresponding trimeric scandium cluster functionalized with aldehyde groups. And you can even translate this to more complex structures. And you can also start applying it to start isolating much complex meta-organic cages. And will show you now one simple example. in which, for example, you can just mix copper and this kind of organic molecule. And when you mix them, you are forming these beautiful green crystals, okay? If you check the crystal structure of this, is the one I'm showing here. This is a three-dimensional structure. And when you mix these two, you are spontaneously forming these co-octahedral cages that are separated by this linker. If you add now olefinic bonds in this position, okay, you should be able to release and isolate the cages. And this is basically what we did. We took these crystals, we put it in a methanol solution, and if you start bubbling ozone, this is what happens. So basically, you start seeing the dispersion, and as you bubble ozone within minutes, you start observing the complete dissolution of these crystals, okay? It's like magic. You're starting dissolving it, and after 10 minutes, you can add ether, you can go home, and the next day, you find the formation of new type of blue crystals. If you now try to solve this crystal structure, you can clearly see that we have released and isolate now this meta-organic cage, this co-octahedral now, functionalized now with 24 aldehyde groups. Okay. So I've shown you that indeed, yeah, we can start playing it with this chemistry and start making the new materials that are very difficult to synthesize. But the next question here was, if we can extend this chemistry to purely organic materials. So can we really start using this clip of chemistry concept to the field of organic chemistry? And in order to prove this, I will show you one example. I don't have much time. And the example I selected is this one, in which you can imagine these are covalent organic framework, which are two-dimensional materials, okay, that are crystalline, okay? It's like a two-dimensional polymer crystalline. And people is always looking at this type of materials as graphene-type materials. But we start looking at it in a completely different way. We start looking them as the repetition of microcycles. You can observe that each one of the pores, in fact, you can visualize it as a microcycle. And these microcycles are indeed separated by an organic linker. So why not now introducing our cleavable olefinic bonds in between these repeated microcycles in such a way that if you do this, you should be able to cleave them and start releasing and isolating the microcycles, being a completely new way of synthesizing quite large microcycles. So basically, this is what we did. In fact, what we start is making these two-dimensional covalent organic frameworks, which are quite easily to synthesize. You just need to take now this organic molecule having amino groups, react with these dialdehyde molecules under isothermal conditions, and you can synthesize this two-dimensional imine link covalent organic framework, okay? And you can clearly see that spontaneously is forming a crystalline framework. Now, you can start applying our concept in this material because you have the microcycles and the olefinic bonds, but we observe that the imine bonds were quite reactive also under ozone. And for this, we decided to do a second step in which we oxidize all these imine bonds into amide ones in such a way that now what we have is this two-dimensional framework linked by these amide bonds, okay? You can clearly see that this is a post-synthetic transformation and that we still keep the same crystal structure after this transformation. Now, in the second step, you just need to apply the ozonolysis reaction. And here, just to remember that when we have a material, we can either use reductive working at conditions or oxidative working at conditions. What it means that if we are successful, now we can synthesize these quite large microcycles having 114 atoms forming the perimeter of the microcycle, functionalized with aldehyde groups, or this other one that will be functionalized with 6 carboxylic acid groups. So basically, this is what we did. Applying the reductive conditions, we synthesized this material, which is this type of solid here, and we ozonize it for 10 minutes, okay? After that, you obtain a completely, so the dissolution of all the solid, in which you can precipitate easily a solid and you can purify. And after purification, you can clearly see that we could form now this microcycle, quite large microcycle, formed by these 6 aldehyde groups in a quite remarkable 90% yield. Okay. And exactly, if you do exactly the same now using oxidative conditions, you can indeed reproduce exactly the same methodology and obtain now the same microcycle functionalized with these 6 carboxylic acid groups. So it seems that indeed you can start applying this for purely organic materials and to synthesize microcycles. And the beauty of this is that you can now merge our clip of chemistry concept with reticular chemistry and use it not only to change the composition of the skeleton, but using the same family is quite easily to extend the synthesis of other type of microcycles. And how we do this is very, very simple. So basically reticular materials say, or reticular chemistry says that if you slightly change one of the linkers, most likely you are going to obtain the same structure. But you are introducing differences in these gray boxes, okay? And this will be translated to the synthesis of the microcycles. So basically, we can now start synthesizing different microcycles just by changing the very simple building blocks that we are using. Let me just give you some examples. Now imagine that instead of this terephtaldehyde, you're using the same terephtaldehyde, but functionalized with fluorine groups. If now you use this one, you are going to make this two-dimensional covalent organic framework. And by using exactly the same conditions, you will be able now to synthesize 2 different microcycles, one functionalized with an aldehyde group, the other with carboxylic acid groups, but also functionalized with all these fluorine atoms. And you can here basically place any different type of functional group that you would like to synthesize. Also, you can do another thing. You can use this to synthesize larger and larger type of microcycles. For example, imagine now that instead of this terephtaldehyde, you are adding 1 extra phenyl ring. If you use this, you will synthesize the same covalent organic framework that will have a much larger pore, which means that you should be able to synthesize a larger microcycle. And indeed, we apply exactly the same conditions in which you can clearly see the formation of this much larger microcycle functionalized with aldehyde group or functionalized with carboxylic acid groups. And you can keep doing it. And you can now, just by changing the very simple molecular building block at the beginning, you can access to the synthesis of larger and larger microcycles. And this is the larger microcycle we have been able to synthesize so far, okay? And with this slide, I just want to finish my talk. I just hope I've slightly convinced you that we are now able to start applying this new chemistry in order to take all the different type of structures that we have in the literature and start synthesizing this new type of molecules and materials using this top-down approach in which we control at the molecular level. And with that, I want to finish acknowledging my whole research group. I think they are the ones who really make all the work that I can show in the different conferences. So I would like to thank all of them. Also, I would like to thank the different people, especially the former members that have largely contributed to the development of this approach, all my collaborators, the different funding agencies that are supporting our research, and all of you for your attention. Thank you very much.
Great, Daniel. Super talk, and what a great start to the symposium.
Thank you.
Lots of interesting chemistry there and a very neat idea, I would say. Now it's open for questions, and just for all of the attendees, the panelists can just raise their hand and ask a question as normal. But for those who are attending, if you wish to ask a question, You could either raise your hand and I will unmute you and you can ask the question, or perhaps what's more easy, if you look down the bottom selection of the slide here in Zoom, you'll see a Q&A function and you can type in your question there. So there's currently no questions there. So perhaps in time-honored fashion, I'll ask the first question if that's all right. What's the limit? You can go up to 162. You know, is there a limit, or do you think you've reached it already? What do you think?
I think that the limit will depend on the solubility of the final microcycle. So with this one we are facing now, we are starting to having problems about solubility, but I think we can add functional groups, for example, in order to try to make even larger one. So the limit, I think, will come more from the formation of the initial covalent organic framework more than the cleavage of the reaction.
Yeah.
Okay.
Yeah.
But indeed, I mean, it can open, in the case of the microcycles, for example, it really opens many different possibilities. We have also started synthesizing microcycles made of polyamides, for example, which are very rarely synthesized using bottom-up point of view. So I think it offers an alternative of making microcycles. And I think the beauty is the high yields we are obtaining. I mean, we are always oscillating between 80 to 90% of yield, which is quite high for this type of microcycles.
Yeah, no, that's important as well. Synthetic chemists, percentage yield still critical. That's true. So again, still open for questions from panelists or any of the 91. Oh, we have a question coming in from Manuel Petroselli. Manuel Petroselli from ISIC. Get your advertisement in there, why not, Manuel? Thanks, Daniel, for the amazing work. I was wondering during your presentation, did you consider the use a photocleavable bond to reach the same goal?
Yeah, this is a very, very interesting question. Yeah. We have started now trying to apply photocleavage in order to extend the generality of our approach. Yeah, you can use, now you can start thinking, this is the first example, you can start thinking using different type of cleavage reaction. The thing, I think the most difficult thing is to be able to cleave it quantitatively. And this is a requirement in our approach. So yeah, but yeah, we are working on it.
Yeah. Very good. Any further questions for panelists? Oh, there's another one coming in. Oh no, that's just from Manuel saying thanks for the answer. So there you go. That was the question and an answer. So that was the answer. Okay, so perhaps in the interest of time, we might move on if that's all right, Daniel. And I know you're off to do a PhD exam. Good luck to the PhD student. And sure, you can watch the video of this later up on YouTube.
Yeah, thank you very much.
Daniel, back over to you, Anton.
Thanks, Daniel, for this beautiful and excellent talk. I'll introduce the next speaker while technical arrangements are made. The next speaker is Professor Arjan Klaijh. Arjan, a good friend. He's performing his research activities at the Institute of Chemical Research of Catalonia, ICIQ, as well as an ICREA research professor. And the title of his talk, as you can see, is photocatalytical strategies to rapidly access molecular complexity. So, Arjan, thanks for accepting, and the audience is yours, I should say, not the floor is yours.
Thank you so much, Anton, for your kind words. And of course, I'd like to join Daniel to thank both Anton and Pat for the invitation, all the others that have been involved setting up this meeting. And I click quickly forward to the next slide, because this is just an informative one. I'd like to spend a few words on the things that we do in my group, ICIQ. So we work on three different themes. The first two categories mentioned over here, carbon dioxide, catalysis and valorization, is basically one of the first lines that we started around 2010. And then soon after that, we also got involved in biopolymer synthesis and engineering, and we are still working on that as of today. But today I'm going to focus on our third research line, and that is what we have dubbed advanced stereoselective. And as you may know, if you want to make complex structures, you will probably very often encounter tertiary or quaternary carbon stereoselective. Just depicted here a few examples of bioactive compounds that contain such carbon stereocenters. And these are, in general, quite complex, let's say, and functionalized molecules. So we always have been admiring these molecules, but also thinking about ways how to mimic that kind of complexity through, let's say, new processes. And if you look at the literature, you see that conventionally there were certain kind of chemistries involved, Diels-Alder chemistry, conjugate additions, cross-coupling reactions, but also among, let's say, one of the first approaches to this kind of complexity was allylic substitution reactions, where you basically make a carbon heteroatom or a carbon-carbon bond. And this molecule, cyanti-Wiggin, I'm not sure if I pronounced it correctly, has been made via a Palladium-catalyzed asymmetric allylic alkylation process. So that shows the potential of allylic substitution in this area. How did we enter the field? Well, we basically, through one of the other research lines, we're making a lot of these carbonates, cyclocarbonates, mostly 5-membered. We have been working on 6- and 7-membered as well, and even 8-membered ones. And this is a very specific example, which we have dubbed VCC, of vinyl cyclocarbonate. So we have here like an olefinic double bond. But more importantly, in the presence of a palladium zero, let's say, precursor, we can decarboxylate, or basically first activate that double bond, and we decarboxylate that intermediate, and then we get access basically to a typical allyl palladium intermediate, an electrophilic palladium allyl. And from here, of course, by ligand engineering, we can steer the regioselectivity and also the stereoselectivity towards either linear or branched allylic compounds. And I have to emphasize here that the use of palladium and other metals like iridium, rhodium, even ruthenium has been reported in this area, is basically the conventional way of doing allylic substitution if you consider the addition of an external nucleophile. Yeah, so this is what we basically tell our students, that this is the classical way of doing allylic substitution reaction. So one example, another example from our group in 2016, we used these cyclocarbonates, aryl amines or anilines, if you like, as nucleophilic precursors, and we were able to make these branched allylic amines. And at that time, if you consider palladium catalysis and, let's say, allylic substitution reactions, this was the first general method to access these kinds of allylic amines. Luckily, we also have here the spanning alcohol, so this allows us to further branch out, the complexity to some follow-up chemistry. We'll not go into that. And here, just a selection of a few molecules that we made, showing the diversity that we can approach. So we have many more examples. And in general, we have quite decent enantiomer induction. Yeah? So typically between 90 and 95%, with some exceptions here and there. So this is palladium chemistry. We have 2 other programs where we focus or zoom in on sustainability, like to do things more attractively, using metals that are, let's say, more abundant, cheaper. So in this, let's say, respect, the use of nickel chemistry was also something that triggered our, let's say, attention. And in a more recent contribution, we have been able to take an allylic precursor. Here we now have a linear allylic compound. And in the presence of this, let's say, this ligand shown over here, this is a pretty complex ligand already, and this nickel 2 precursor, we can basically induce a manifold that involves the initial reduction by zinc. So zinc is here a metallic reductant. We reduce the nickel 2 to nickel 1. We get oxidative addition, going then to nickel 3. Then we do a single electron transfer reduction. So we get back to nickel 2. And now basically we set the system up for nickel 0, nickel 2 redox couple, which is basically mostly the productive cycle that we have in this manifold. I don't have the time to go through all the mechanistic details, but they can be found in the reference shown over here. More interestingly, maybe from an organic point of view, is that we find here basically a head-to-tail arrangement of these allylic groups. So these are 1,5-dienes with this ordinary carbon stereos. And compared to the literature that was reported up to that moment, we had a different kind of, let's say, arrangement of these, let's say, these two allylic groups as branched or head-to-head and tail-to-tail type of 1,5-dienes have already been reported. So the good thing is that we can produce a lot of interesting molecules. So this is what you can call formally a cross-electrophile coupling between two allyls. And although we use mostly this ligand over here, so this is a ferrocene-based diphosphine, in some cases we found that other kinds of ligands, stereically demanding ones, based on the SECPOS family, even gave a slightly increased in angioinduction, although in other cases, as shown over here, we found basically that we got better in angioinduction, but a lower regio control and therefore also a lower yield of the desired product. So this basically shows that there is no perfect ligand to run an entire scope of products. Stepping away a little bit from, let's say, metallic zinc as a reductant, so that is not maybe the most sustainable way of doing reductive chemistry. We thought about photoredox. I mean, most of you probably are aware that photoredox chemistry as a way to produce reductive equivalents is a very attractive field. There are a lot of people working in that area. And one of the initial ideas was to do alylation of aldehydes. That is a process that has been studied by many people. Basically getting to these kind of intermediates, which then lead to linear or branch products. So you need to control basically this equilibrium. And more importantly, to control these transition states, which are formerly known as Zimmermann-Traxler type of transition states. That controls the diastereoselectivity selectivity, at least in this particular example. So at the time, basically there were two ways of doing this alylation of aldehydes. One is the crucial alylation, and the other one is the use of a metal and a photo catalyst, so basically to do metallophotoredox catalysis. And there were actually at that time there was a lot of work already done on chromium, titanium, and nickel chemistry, and a few, let's say, examples of metallophotoredox involving cobalt catalysis. But more importantly, in terms of the organic chemistry, essentially, if you look at the, let's say, the alylation procedures, typically large excess of allylic reagents were used and poor diastereoselectivity was reported and in quite limited scope in terms of, you know, the organic molecules that you can access via these former methods. So we thought that we could do maybe something interesting using or combining cobalt catalysis with photoredox catalysis. And so we started to use our vinyl carbonate and to combine it with an aldehyde. This is benzaldehyde, so this is our benchmark case. And this is the standard product that we investigate. And finally, we'll quickly go through the details. The ligand screening basically showed that this DPE-phos, which is a pretty simple and cost-effective ligand, already gave quite efficient catalysis, so good yields, reasonable diastereoselectivity control, and good substrate conversion. And as a photoredox catalyst, the most effective one was this 4ZZ-IPM, which is an organic based photoredox catalyst. So that is also attractive in the realm of maybe the whole catalytic process to make that, let's say, as sustainable as possible. So the scope of the products is actually quite broad. So we have more than 40 examples, or here it says 40 examples, where we can basically vary the carbonate and the aldehyde. And I've depicted here a number of examples, not just to impress, but also to show some of the limitations that we have. So some limitation is with aliphatic aldehydes that is shown over here. We get basically lower yields, but overall we get quite decent diastereoselectivity. So 9-to-1 is quite typical in this, let's say, process. Okay, this is a very complicated slide. I apologize in advance, and I will simplify that in a second. But I would just like to show you the great work that Bart, at the time a postdoc, but now at the University of Barcelona, also an ERC grantee, he did a fantastic job in basically investigating the details of this, let's say, ladder process. So the formation of these 1,3 diols through cobalt photoredox catalysis. So he looked very much at the photocycle shown over here, and also in great detail of this, let's say, cobalto cycle. We'll now simplify this into basically the cobalt cycle. And so we know that we started with a cobalt-2. We have an X-ray analysis of that compound. It's very simple. And now we do a single electron transfer, let's say, based reduction. So we end up then with this cobalt-1, which was further investigated by spectral electrochemistry. And then we can do an oxidation addition. We can follow that by cyclic voltammetry and some other spectroscopic techniques. And now this cobalt-3 undergoes this slight rearrangement, a single electron transfer to end up here with a cobalt-2. And now in this cobalt-2, we can form a single non-Taxler type of intermediate. And here is where the diasteric selectivity is controlled. Yeah, so we see, for instance, in this particular example, clear energetic difference, so delta G. And by DFT, we know that we can get the diasteric control around 9:1, which nicely aligns with the experimental, let's say, observations. So this was, at that time, I think, maybe a very detailed analysis of both the cobalt cycles over, let's say, the photocycle. And it showed also some of the limitations when adding more, let's say, cobalt to the system, which basically resulted in lower catalytic efficiency. That is counterintuitive, and Bart made a fantastic job in order to explain let's say, the reasons for that. So far cobalt and combined with photoredox. What about other metals? So nickel has been reported also quite often to combine with photoredox catalysis in a formal dual approach. And in this way, you can design interesting multi-component type reactions. And at that time, we believed that this would create new impetus for what we call allylic complexity. So at the time, of course, a few approaches were already known to get access to allylic compounds, but mostly based on 2 components, and for instance, based on these kind of allylic precursors, so these vinyl epoxides. But their synthesis is much more limited than the vinyl carbonate, so that is one particular advantage. And in this particular setup, we were able to combine 3 components, and we'll see later on how that works, let's say, from a mechanistic point. In this process, we can control the regio and stereochemistry actually quite nicely, and we can make elaborate quaternary carbon centers, as you will see on the next couple of slides. So Tian, who is graduating in December, took these vinyl carbonates, these trifluoroborates, and these diaryl 1,1 diaryl, let's say, olefins. And I will not go through all the details how we optimized it. It was a lot of work, but finally we were able to get these kind of compounds. Yeah, so where we have basically all 3 components incorporated into the final structure. And now you see already that it can become reasonably complex. So by variation of the vinyl carbonate, which you see over here, different options to introduce, let's say, complexity at different positions in the molecule, we can do the same if we vary the trifluoroborate. Yeah, we can have tertiary radicals, secondary, and even primary ones. They work to a lesser extent, but you can see that you can introduce actually quite complex, bulky type of substituent or substituents and cyclohexyl or cycloalkyl, and also, linear ones. And thirdly, we can, of course, vary the arrow groups here. As you see over here, you can use 2 different arrows, one pyridyl, one simple arrow, or here, protected alkyne, 1 arrow, and here we have an ester and an allyl combination. So this is very nice. It shows that we have a lot of opportunities here. We did a number of controlled experiments. And we also explored the synthetic use, which is shown over here. So if you take that molecule, you can, of course, make very simple protection of that alcohol group. You can oxidize it to get access to this alpha-beta unsaturated aldehyde. We can access allylic amines, which have their own merit in organic synthesis in a very simple way. And we can oxidize very, I would say practical approach and get these tetra-substituted epoxides. If we take these reagents and we treat them under the, let's say, standard conditions, so we have 4ZZ-IPM, which is the organic-based photocatalyst, we get here basically the Z-configured product, in about 9:1 diastereoisomeric ratio. If we then change for the iridium photocatalyst, we basically revert the stereochemistry. So we get the same product, but we get the E-isomer. This shows very nicely by changing simply the photocatalyst, you can access both stereoisomers to a certain extent at least as the major component. In order to give credit to the occurrence of radicals, we used very standard tempo addition that shut down catalysis. If we take the nickel complex out, we basically show that it's crucial to have that part of the catalytic, let's say, approach, because if we don't have the nickel there, we basically form a byproduct. So it shows that we can still form radicals, but the radicals then basically, or the radical formed from this trifluoroborate combines with this radical acceptor, and then we get a hydrogen transfer to finish out that reaction. So we need the nickel essentially to run these three component reactions. If we look at the standard conditions, we use basically this acid, this phosphate acid or hydrophosphate acid. We also use other protic donors. What we see is that we get more or less similar yield of the final product, showing that the acid basically acts as a proton donor towards the end of the reaction. So here I'm showing you the mechanistic, let's say, proposal that we have based on a number of control experiments and observations. So basically, we start up here. We do it here at the nickel 0 level. We do an oxidative addition. We get excess CO2 and nickel 2 intermediate. And now two things can happen. One, we get a single electron transfer reduction forming basically a nickel 1 and then a reductive elimination to form basically here this radical species. So we reform the nickel 0, so we can then continue this cycle. And this radical then combines with another radical that is formed through the photo cycle, where the photocatalyst basically reduces the trifluoroborate to form this tertiary radical that then combines with this diaryl olefin to form this secondary radical, which now combines in a radical cross-coupling. And we have proved that this kind of radical indeed is there as an intermediate. However, we can of course not exclude the radical metal crossover. So where the nickel 2 complex combines with the radical, forms a nickel 3, then undergoes a reductive elimination and a single electron transfer to form the final product. And what we believe is it basically depends on the steric, let's say, modulation that you have here between the R group and the LL, and of course the ligand. Which of these two mechanisms is probably the dominant one? Then to extend this approach, what we did, we were actually aroused by the fact that, substituted olefins, basically with 4 different carbon substituents, is an interesting target because there are a lot of, let's say, bioactive compounds out there that have this motive. And we thought that radical chemistry could help us to achieve that objective. Up to the point that we started to use radical chemistry in order to access olefins. There were, of course, some work. There was some work that had been done already by the Shia group, and Macken also reported a very interesting, let's say, approach in Nature Catalysis in 2019. And these examples are given over here. One of the challenges still that was out there was to make tetra-substituted olefins, and do that in a stereoselective manner. So this was basically our starting point, and here you see again back our star compound, these vinyl carbonates. And the idea was to form a radical that would add on to that double bond, as you can see right here, and then induce basically a decarboxylated double bond relay. So that is visualized over here. So if we basically reorganize the molecule and decarboxylate it, we should form this intermediate radical where we now have that double bond going or being basically relayed to the internal position. So this is a CO2 extrusion and double bond relay type of sequence. Then we get a single electron transfer reduction, so to form this alkoxide, which then, in the final step picks up basically a proton and forms the final product. I will not go into detail, but here it's very important to match the polarity between your radical and your radical acceptor. And another thing that I can mention is that this decarboxylation step is quite important. Okay, so we thought that the formation of alpha minor radicals, so here we have just a pyrrolidine, which we can basically use as a radical precursor. We form then a radical at this position, and then we induce the radical addition, and the entire manifold will then automatically go towards the final product under these optimized conditions. And we found that the iridium photocatalyst was the best photocatalyst for this approach. So we need a iridium photocatalyst, but we do not need an additional transition metal to control basically let's say, stereochemistry here in this manifold. So how does it basically work out? Well, actually, you can use many different, let's say, amines as radical precursors, as you see over here in orange. Quite complex type of amines, such in this case, or maybe this particular case. And you can also use a great, let's say, variety of vinyl carbonates, either being substituted at this R3 position or at this arrow. You can scale up. Scale up in photocatalysis, for those that are familiar with photocatalysis, is not necessarily easy. So here we scaled it up to a sub 400 milligrams, which is not too bad. You can extend the approach by using other heterocycles. They also work. So you can use a cyclic carbamate, or you can use a keto carbamate. That is shown over here. It also works. You get good yields, good stereo control. Here we basically elongate this chain by an additional carbon or a methylene fragment by using a 6-membered vinyl carbonate. And here we have an example of a keto carbonate where we finally then make here an allylic amide as a final product. Other radical precursors can also be used. So trifluoroborates instead of these these amine or let's say amine precursors can also be used, or trimethylsilyl protected amines. And this I will show you in the next slide. Also has interesting applications. It also works basically good yields for radical-based reactions and excellent stereo control. Here you see why it's important to have different kind of radical precursors. So if you have amine-based drugs, which are equipped with this TMS group, now you can connect this allyl group, this olefin, if you like, to these drugs. So this comes from amoxapine, this from cytosine, and it works basically in a similar way with good yields and generally, except for this particular case, also high stereo control. What you now further can do is make drug hybrid molecules. So here we have basically this intermediate, or this is a different one. Now we can here do a Steglich, let's say, esterification reaction with indomethacine, and now we have this hybrid drug molecule based on this protocol. So far, I discussed 2 component reactions. What about 3 component reactions, and how to use them productively in order to create, let's say, additional chemical space for those that are, let's say, interested in using small molecules to do drug discovery and drug development. So we thought that the use of these propylanes here, basically, in this molecule, it's a bicyclic pentane, so it's a BCP, but you start off with propylene itself. These compounds are quite interesting because there are a number of these compounds incorporating such a unit that have been reported to be under investigation for their pharmaceutical, let's say, activities. And we thought that we could try to do something, similar, or at least something that is quite useful, because at the same time, these VCPs, they are biasosteric replacements for parasubstituted aryls. And allyl benzenes, there's been a recent review on allyl and how to build up, let's say, bioactive compounds from allyl. So we thought that that would be an interesting approach to create new chemical space and new opportunities. So again, here we have the carbonate, the three components, and to form these kind of compounds. And the interesting thing here is that we can not only introduce directly an alylic alcohol, but also an alylic amine, basically form enines, enamides. And I just want to quickly show you a couple of things that we can do. So this is all mostly stereo-defined. You can see that by changing the carbonate, we have access to a lot of complexity. We can do the same for the sulfone that we here introduced, which is by itself already a very interesting, let's say, a motif for drug development. It can also be varied quite easily. And I just need 1 or 2 minutes, hopefully I'm allowed. And as I said, so we can introduce alylic carbamates as well in one step, alylic amines. We can use azide or azide precursors to make azide radicals that then combine with the other reagents in this protocol, and also CO3 radicals. So that further is very nice. Then just to show you that we can do some post-synthetic synthesis, so we can do a formalize into reaction onto this, let's say, alcohol. We can also do click chemistry. We can use a three-component reaction in order to produce, let's say, pharmaceutically interesting compounds and make hybrids, as I've shown before, in the two-component reaction via Stecklich esterification. This is really the final slide. Few control experiments. The 2CR reaction actually takes place if you take out the propylene. Then second, if you use another, let's say, radical acceptor, in this case styrene, you basically get 2CR selectivity. So the propylene is very, very important in this case as a radical acceptor. And if you take out the aryl substituent, then basically you lose selectivity. And that shows that the carbonate also is important. Just a final comment, which is that the sodium sulfonate that we use at the beginning of the reaction is basically the most efficient quencher of the excited photocatalyst, as the other two components hardly quench at all. So that is how basically we start the reaction. I'd like to here finalize by 2 take-home messages. Hopefully I've shown you today that multi-component strategies are available now to induce, let's say, the formation of complex alylic syntones, and hopefully useful in pharmaceutical fine chemical development, and that the use of photocatalysis offers, let's say, at least in some cases, a cleaner and more convenient way than maybe conventional, let's say, alylic chemistry. Last but not least, many thanks to the people. This is a recent photo, the funding agencies, the people that have been involved at some stage in the collaborations, and I thank you for your kind attention.
Super, Arjen. Great talk. Wow, that's very advanced stereoselective synthesis for early on a Friday morning. Congratulations, and nice to see the decarboxylative asymmetric allylic alkylation playing such a big role in a lot of the work that you do. So again, for those panelists who wish to ask a question, just raise your hand. Same for those who are attending. If you wish to ask a question, you can either raise your hand and then I will unmute you, or you can simply type your question into the Q&A box, and you can see that down the bottom of the Zoom slide. So again, please do that. So Anton, you're first in there, please.
Yeah, excellent talk, Arjen. Outstanding results. My question was, what's the potential that you see in all the new building blocks that you have so efficiently synthesized to be incorporated in new drugs? Yeah, not.
No, yeah, that's an excellent question. I think, from my point of view, that the highest potential that these methods have is to maybe functionalize existing drugs. That you need, of course, then particular fragments to work with. But I think that is the most interesting option that there is out there. Also, the last chemistry that I've shown, the three-component reaction, there are a lot of, let's say, BCP. There's a lot of BCP chemistry going on, these bicycle pentanes. And some of these, as I've shown, are already, let's say, being investigated for their let's say, biological activities. But another interesting thing that we discovered recently is the interesting, let's say, or the important role, maybe, if I may say so, of allyl benzenes. And so if you, for instance, think about eugenol and eugenol derivatives are now being investigated for various, let's say, purposes, anti-cancer, even anti-cancer agents. So that is where we think this chemistry could lead to. Yeah, that's the short answer. I mean, we can always discuss in a more elaborate way, but I think that is where we should go with this chemistry.
Great. Ariane, perhaps I could ask the question on your cobalt photoredox catalysis mediated combination of the vinyl carbonates ultimately with an aldehyde. So you're getting very high levels of diester selectivity. What about an anti-selectivity?
Yeah, that's a good question. I mean, we have a slight practical problem there. So we also investigated in brief an anti-selective version. I don't think it's impossible, but it requires quite a lot of ligand engineering because the ligand is quite important. So we tested really a lot of ligands. Typically what you see some kind of decay in efficiency, in stereo induction. Basically what we see is that DPE-FOS works really well for the diester selective version, but when you change a DPE-FOS for other diphosphines, it basically shuts down catalysis. So there is something with that ligand. So I'm not sure if we can mimic in a simple way the DPE-FOS, turning, or turning the diester selective process into an anti-selective process and identifying a ligand that will help us to retain the activity, but, with the option to induce stereo selectivity. So the short answer is, yes, we tried, didn't work out, and mostly because we haven't identified the ligand yet in order to do so.
Yeah, no, fair enough. And actually, Bart Limburg, who did that work that you highlighted there, he was a attendee at the Young Investigator Workshop in Dublin earlier this year, and he was selected as one of the 2 people to represent the division at the ACS meeting in Denver, as it turns out. So there's the connection.
He did a fantastic job in developing the chemistry here when he was here, and now we can build on that basically in our group to further explore these kind of processes.
Now, we have another question. Manuel Petrocelli is wide awake and very inquisitive this morning, again from ISIC. Thanks, Ariane, for the interesting talk regarding the ACS catalysis paper from Quinn published in 2023. How do you explain the formation of traces of product from a radical-radical coupling in the presence of TEMPO in the reaction mixture? Do you not expect that the quenching of the 2 carbon-centered radicals by TEMPO, essentially under diffusion, is much faster compared to the radical-radical coupling reaction?
Yep, good question, and not an easy answer. Remember what I said about the mechanism. So I'm not a believer about absolute mechanisms. Sometimes when you read literature, people say, well, this is the mechanism. I say, well, it's a mechanism for that substrate combination. So what I said is that we believe that there are 2 possible mechanisms. So one is radical-radical coupling, and the other one is the formation by a radical metal crossover to form a nickel-3 intermediate and then do a reductant elimination. So the radical can be saved, so to say, by forming a nickel-3 complex. And if that nickel-3 complex is not interacting with the TEMPO, it could still form product. But it really depends, I think, on the substrate. So every substrate probably has a preference, for one of the 2 possible mechanisms. And when you shut down one mechanism, it doesn't mean that the other mechanism is completely shut down. So maybe in that particular case, that is just me thinking out loudly, we still have, as a minor, let's say, type of mechanism, the one that goes through the nickel-3 intermediate. And from there, we could form a little bit of product. So maybe that could be an explanation, but we don't have, let's say, absolute proof. What I believe is that probably on a substrate combination basis, we can have the 2 mechanisms with, for each substrate combination, a preference, or maybe, so you have a minor type of mechanism and a major one. And depending on the steric requirements, I believe that you can push towards one mechanism or the other. That is, I think, the best answer I can give now.
Great. Good, Ariane. So perhaps now, again, just see if there's another Q&A coming in. No, got it. Thanks for the answer again. Thank you. So I think in the interest of time, we might pause there if that's all right, Ariane. Again, super talk. Back over to you, Anton.
Thanks, Pat. The next speaker is Carme Rovira. She performs her research activities at the Department of Inorganic and Organic Chemistry of the University of Barcelona as an ICREA research professor. The title of her talk is Protein Glycosylation Mechanisms. Carme, thank you.
Thank you, Anton.
The audience is yours.
Thank you, Anton. Do you see the presentation? Everybody sees the presentation?
Yes. Yep.
Yeah.
Okay. So thank you very much for the invitation to this interesting symposium. So I'm going to talk about protein glycosylation mechanism, which is about the binding of carbohydrates to proteins, which is one of the most important protein post-translational modifications of proteins. I'm going to talk about this and how do we model this with computer simulation. So when we talk about carbohydrates, about sugars, one normally thinks about this type of sugars in our diet, saccharose, starch, lactose, which is the sugar of the milk, etc. But there are other type of sugars, other type of carbohydrates that are maybe less known, which is the carbohydrates that are attached to our proteins and also our cells. This is an image of an electron microscope image of an endothelial cell, and you can see all this type of hair here. Those are sugars. Those are carbohydrates bound to the membrane of the cell. They occupy a very big, large region of the volume of the cell. So there is a large sugar forest that is covering our cells. This is the so-called glycocalyx. The glycocalyx is about between 10 and 100 nanometers thick, and in this region we can see sugars, carbohydrates attached to proteins that are embedded in the membrane. Also, other proteins are also glycosylated proteins that are not in the membrane. There could be carbohydrates bound directly to lipids of the membrane. This is the so-called glycolipids. Or there could be also this type of proteins that are like a long polypeptide, which is heavily glycosylated. Those are the so-called mucin proteins that appear in our mucosas. And these carbohydrates here are very important to feed our microbiota. So there's a lot of carbohydrates covering our cells. And they are not there just for decoration. They play a major role on how cells communicate one each other. They are like antennas that the cells use to communicate one each other. And by doing this, this carbohydrate bind to, for instance, receptors in other cells. No doubt that how cells communicate. And then by doing this, but through this communication, through these carbohydrate antennas, they are also protecting us from invaders like viruses or bacteria that are trying to interact and penetrate into the cell. The carbohydrates have this protecting role. This is how they are represented. I don't know if you see the titles of maybe I put it here. This is how are they represented, typically with this kind of Lego-like structure, and this is with different color codes. Every monosaccharide is a different color or a different shape depending of the type of substituents on the sugar. So when these glycoforms are defective or they are not assembled properly, many problems can occur, many diseases, because then cells cannot communicate well each other. For instance, one can get I don't know, autoimmune diseases, asthma, allergies, a lot of different type of diseases, and even cancer. It's been shown that cancer cells are also covered by sugars like normal cells, but they have a different sugar pattern. For instance, some of these carbohydrate trees are more complex, so they show some aberrant glycosylation, and some of the carbohydrates are smaller; they are truncated, and this is used nowadays to diagnose certain types of cancer, like pancreatic cancer. And since this was discovered, now is several years ago, many years ago, it's been a lot of interest in trying to understand how the many enzymes that are involved in building these glycans, enzymes involved in the post-translational modification, how these enzymes work, because some of them are overexpressed in cancer and it's necessary to block them. And I will tell you in this talk, I will tell you about some of the recent work that have been done in some of these enzymes. One is about one enzyme is MGAT5. It's an enzyme that is responsible for catalyzing the formation of a glycosidic bond between this N-acetylglucosamine and this mannose sugar here. So we after this enzyme, then the sugar, the glycoform can grow, and this is mainly present in cancer cells. Another enzyme that I'm going to talk is about GalNAc-T2 that is responsible for catalyzing the formation of this glycosidic bond between N-acetylgalactosamine and a residue of a protein. So let's go for the first one. Or maybe I show you first also how to model this type of, how do we study this mechanism of enzymes. So first we need a crystal structure of the enzyme. We need a structure or even an AlphaFold model we can also use. And what we do is that in the field of enzyme catalysis, one normally uses the so-called quantum mechanics, molecular mechanics approximation. So we need a model of the enzyme, and we cannot treat all the atoms by quantum mechanics. This will be necessary to describe a chemical reaction. You need quantum mechanical accuracy, but we cannot do such a big system, quantum mechanics. So fortunately, there is these methods called quantum mechanics, molecular mechanics, hybrid methods, in which we treat one part of the system where the reaction takes place at the quantum mechanical level. Normally, this region is small, is the active site. And then the rest of the protein and the solvent, for this, we use a more approximate level. We use a force field. We use normal molecular mechanics. On top of this, we combine this with molecular dynamics, which is less common in the field. So what we do is QMMM, molecular dynamics. So we move all the system to at room temperature. Still, this is not enough to describe a chemical reaction in an enzyme, because in a chemical reaction, you are in a potential well, in the reactants well, and you need to travel to the products to describe the reaction. You need at least a trajectory, one event in which you go from reactants to products. And this doesn't happen normally in a time scale of the simulation. That doesn't happen in the time scale of molecular dynamics, because there is a barrier to be overcome. And the time necessary to overcome this barrier is too large for the computational power nowadays. So, and it will be like this for many years, I'm afraid. So it is necessary to couple this methodology with another one that pushes the system over the barrier. And let's say in a way to speak, there are several methods that can do this. The one that we use in my group is metadynamics. That is very efficient to go from to activate rare events, in particular chemical reactions. It's based on the definition of a few collective variables that normally they are distances involving the atoms that are atoms of the bonds that will be cleaved or formed during the reaction. And then with this method, then we can go from reactants and products and also backwards. So this is only a glimpse of the methodology that I wanted to mention. Now I go to the reaction that to the enzyme that we were investigating. So the first enzyme that I want to show you is MGAT5, so-called N-acetylglucosaminyl transferase 5. MGAT5, what it does, it binds N-acetylglucosamine attached to UDP. UDP sugars are typically transferred not alone, but they are attached to a nucleotide, and then the nucleotide is released during the reaction. So the enzyme transfers N-acetylglucosamine to an acceptor, a glycan acceptor that is this glycoform that is partially made. So it adds a new sugar to this glycan, to this glycoform. And it does it with inversion of the so-called inversion of the anomeric configuration. MGAT5 is a very important enzyme in biomedicine. It has a biomedical relevance because it strongly drives cancer when it is overexpressed. So it's been found in certain cancer, like colon cancer, that this enzyme is overexpressed. So it's necessary to find inhibitors for it, molecules that can block the enzyme. And for this, we need to know what is the mechanism of the enzyme, first of all. And unfortunately, there was no crystal structure until a few years ago of the enzyme. And one of these crystal structures were done by Gideon Davis in the University of York, that is one of our collaborators. And he could get a structure with the donor, with the UDP, and part of the acceptor. As you can see here, this is the 3 mannose residues, these mannose sugars here. This is the glucnag that is here. This one was also in the experiment. They also put it, but it was not seen in the crystal structure, so we didn't use also in the modeling. And the complete structure was not there because actually this sugar was missing, the sugar of the donor, so we had to model it. And we model it, take into account the position of the waters in the crystal structures. We thought that the oxygen of the water molecules will be in a similar position as the hydroxyl of the sugar. And that worked well. We got a very good Michaelis complex. It's quite stable and molecular dynamic simulation. And once we have the Michaelis complex, then we can see, then we can closely inspect. And then what we see, for instance, is that there is the hydroxymethyl group of this mannose that is supposed to be the nucleophilic mannose is quite close to the anomeric carbon. It was about 3.5 Armstrongs and very well oriented for an SN2 reaction, very well oriented to do the nucleophilic attack while the UDP is the living group. We could also see that there is a residue of the enzyme, a negatively charged residue, glutamate, that is quite close to the OH, very well oriented to extract the proton during this nucleophilic attack that is assisted by this residue. This residue is essential, so mutation of this residue inactivates the enzyme, so that confirms that this is probably the catalytic base of the reaction. Then we initiate the modeling using QMM metadynamics. And you can see how this works. That these simulations were done by Beatriz Piniello in my group that is going to defend the thesis next month. And maybe it doesn't work now. Sorry, I think the movies are not working. Strange. Let's see. No. There should be a movie here. No. Anyway, sorry for this, but there should be a movie in which you see that the OH attacks the anomeric carbon, and then this group departs, and then you have the glycosylated mannose. This is the transitional state of the reaction in which the two bonds are partially formed and partially broken, and the proton has not yet transferred to the glutamate. So with this structure that we get from the reaction, important thing to see is the conformation of the sugar at the transitional state, because it's the type of half-chair conformation. This is the conformation that should be targeted to design inhibitors for the enzyme, inhibitors that block the enzyme. And after we did this study, and a group of chemical biology from the Francis Crick Institute contacted us because they were working on this enzyme, and they were designing enzyme mutants that could accommodate modified donors with a voluminous group that could have a clickable tag. And together with them, we helped them to design this type of mutants, and they found out that mutating two residues in the active site and then using as a substrate a sugar substituted by porous acidobutyramide, they could label the enzyme. They got very good results in vitro. They could label the mutant enzyme, so they could use this to attach a modified sugar in the glycans. And this modified sugar can be used now with click chemistry to accommodate other groups. So this can be used to label these glycans in cells. So Another enzyme that I want to discuss with here is GalNAc-T2, N-acetyl galactosaminil transferase 2. GalNAc-T2, what it does, it is attaches an N-acetyl galactosamine to proteins, in particular to serine or threonine residues in proteins. So that this initiates glycosylation. It attaches the first sugar to the protein. And this enzyme is quite important because it catalyzes the formation of this glycosidic bond here. So it forms the TN and so-called TN antigens, which are a hallmark of cancer cells. In that case, the protein is a bit more complex than the previous one. It has two domains, a catalytic domain, this has been shown through X-ray structure, a catalytic domain, an lectin domain that is supposed to be important to attach to the protein to be glycosylated. And they are connected by a linker that supposedly is quite flexible. So there was crystal structures of this since several years ago, but there was no crystal structure with the substrate inside so that we could use for the modeling. Then our collaborator in the University of Zaragoza, Ramon Hurtado, could get a structure with the donor here, with the UDP GalNAc, and also acceptor peptides, so peptides that can be glycosylated by the enzyme. This is one of the structures that he did, in which the acceptor is bound to the lectin domain. That's still not so good to initiate the reaction because the acceptor is far from the donor. So there should be there's a sugar here that should be attached to this peptide, but it's quite far. Then by doing molecular dynamic simulations, what we saw is that the enzyme has well, again, this motion doesn't work. The movies don't work. Maybe I can put it like this. No. Maybe I can do like this. I don't know. This is the motion that the protein has. We saw it with molecular dynamics. So the linker is really very flexible, and it helps to approach the peptide to the catalytic side. Once the peptide approaches is close to the catalytic side, then what we see is that a threonine of the peptide is very close. The OH of the threonine is making a hydrogen bond with the UDP of the donor. That's a very good configuration for sugar transfer with retention of configuration, which we know is what happens in this enzyme. So this is different from the previous one. The previous one, there was inversion of configuration. This is rotation. And this type of reaction, sugar transfer with retention of configuration, has been controversial for several years. We contributed to solve the mechanism 10 years ago. And what we confirm is that they go through a mechanism called front phase, because the reaction takes place in the same phase of the sugar, below the sugar, in the alpha phase. And it takes place without intervention of any protein residue. There is no catalytic base residue like in the previous case, like in inverting glycosyl transferases. In that case, the catalytic base actually is the phosphate, because this oxygen is the one that is going to take the proton of the threonine, and is the nucleophilic attack. Then it can take place on this region of the space. You can see how the reaction goes here. You can see this bond will break first. So it's a two-step reaction. Now we form an oxocarbenium ion that we see because there is a small minimum in the free energy landscape. And then it collapses towards the threonine while this proton is being transferred. So that's the glycosylation of the threonine via a front phase reaction mechanism. And this front phase reaction mechanism, we have also model and confirming other enzymes, not only in protein glycosylation, but also for the formation of glycosidic bond between when the acceptor is a sugar, like in trehalose 6-phosphate synthase, and also in glycogen, in which US is a glucose formation of bonds. So let me put this again like this. So a more recent work that we have done is about N-glycosylation. N-glycosylation is the attachment of sugars to asparagine residues in protein. So in the previous case, it was a sugar attached to serine or threonine. Now it is, I'm talking about sugars attachment to asparagine. This is the second most abundant post-translation modification most of proteins. The first is O-glycosylation, and the previous case, the second one is N-glycosylation. So maybe you heard about the SARS-CoV-2 virus and the spike protein of the SARS-CoV-2 virus that is full of carbohydrates around. These carbohydrates are N-glycans, are all attached to asparagine residues in the protein, for instance. So there is a very important enzyme that catalyzes N-glycosylation, which is N-glycosyltransferase. So it was recently, 4 years ago, reported that bacteria can also catalyze N-glycosylation, and they catalyze a very simple N-glycosylation reaction. There are enzymes in bacteria, NGTs, that bind UDP glucose and transfers the glucose to asparagine, just the glucose, instead of the other enzymes that are more complex that we have in humans, that what is transferred is a complete oligosaccharide. So that discovery was very important, because since we don't have this type of enzymes, targeting these enzymes, inhibiting these enzymes could be good to fight infections. Actually, Haemophilus influenzae also has NGTs, and the Haemophilus influenzae is a very pathogenic bacteria. So, but so far, at the first year, there were only small, very simple crystal structure with practically nothing inside. So nobody could know what is the mechanism, because the donor and the acceptor had not been solved. And there was some hypotheses about how this reaction could take place. This is the donor, the glucose with UDP. This is the asparagine to be glycosylated, the asparagine of the protein that is being glycosylated. So initially one thinks that the nitrogen attacks the anomeric carbon, and then the sugar phosphate bond breaks, and there should be some protein residue that takes the proton of the asparagine in this process. But this protein residue could not be identified by mutations. Nobody knew who was this protein, this catalytic base of this reaction. Then Ramon Hurtado in the University of Zaragoza could get a new structure in which there was the acceptor there, an acceptor peptide that needs to be glycosylated, and part of the donor, there was the UDP. So we reconstructed what is the missing part. We added the sugar here. We used several strategies like docking the whole glucose UDP, or adding just the sugar, or doing simulation with another structure in which there was a galactose instead of a glucose. But independently of the strategy, we arrive to this type of configuration in which we have here the UDP glucose. This is the asparagine to be glycosylated. We're quite confident about this because this UDP glucose is in the same pose as in another enzyme of the same family, OGT, that catalyzes a different reaction, but is from the same family, so it's similar structure. So this other enzyme, UDP glucose, it can be practically superimposed to this. So what we see here is that actually, as it had been described in experiments before, we couldn't pinpoint any protein residue around the asparagine that is able to unprotonate the asparagine. There was not a residue here, really, independently of the snapshot that we take during the simulation. But we could see that there is a hydrogen bond between the amino group and the alpha phosphate of the UDP. So we thought, okay, maybe the alpha phosphate, not the beta as in other glycosyltransferases, is the catalytic base. Maybe this proton transfers here, and then is how the amino group can attack. So let's try this. And then now it works. Okay, so we tried this simulation, we tried this, pushing the asparagine towards the anomeric carbon, and then it does the reaction. So it glycosylates the asparagine, and it's proton transferred to the beta phosphate, to the alpha phosphate, sorry. So that looks fine. However, if you look at the free energy landscape, and then what we see is that the energy barrier is very high. It's more than 750 kilocalories per mole. That means this reaction, as it is unfeasible. It cannot take place. Something is missing here. And we tried, we did many trials with different collective variables, but we always got this high energy barrier. And we think this is because the attacking angle is very close for an SN2 reaction, because this is inversion of the anomeric configuration. If this guy here, this amino group, needs to interact with the alpha phosphate, and at that time, the same time approach to the anomeric carbon, that means this angle is very small. Then what happened is that by serendipity, in one of our simulations, that what happened is that a proton of the amino transferred to the phosphate and then to the carbonyl. So we got a tautomerized asparagine, a tautomeric form of the asparagine. We go from amide to the imidic acid. We then realized that from the imidic acid form, from the tautomeric amide, this angle is more open. So maybe that will be more favorable for attack because it's a larger angle, because we are gaining like one covalent bond here. It's still making this hydrogen bond, but this angle of nucleophilic attack is larger. So we tried this. There's new simulation from the tautomeric state of the asparagine, and it also works. So the asparagine can attack the anomeric carbon, and this proton is transferred to the alpha phosphate. So we reach the same product as before, the glycosylated asparagine, but now the energy barrier is much lower. It's about more than 20 kilocalories per mole lower. So we think this is the mechanism for N-glycosylation in NGT. This is our proposed mechanism in which the phosphate acts as a catalytic base, and the asparagine uses the imidic acid form for catalysis. So this is we are doing further work on it now, trying to design inhibitors for the enzyme. And this is what the three examples that I wanted to show you for protein glycosylation. And so in summary, what I've been showing you is that glycans decorate our proteins and cells, and they play a major role in health and disease. That enzymes that process these glycans can be therapeutic targets, and I hope that I convince you that these type of simulations can unveil the mechanism, the intimate mechanism at molecular detail, in synergy always with experiments, of course. And let me just thank the people that has been involved in this work. This is my group. I talk mainly about work by Beatriz Piniello, that is going to defend thesis in next month. Laura, that is a new PhD student that is continuing the work. And I talk also about work from former members that are around the world. And I would like also to thank our international collaborators, experimental collaborators. So, and all of you for your attention. Thank you very much.
Thank you very much, Cara. Excellent presentation on glycochemistry, glycans, quantum mechanics, transition state mimics, lots of things going on there. So again, open for questions from panelists or attendees. And again, to remind any of the over 100 attendees that are currently online, thank you again for attending. There's a Q&A button on the bottom of the Zoom slide. You can type in your question and I'll read it out, or you can raise your hand and I will unmute you. So again, that's open for questions. Carmen's presentation is open for questions. So I suppose a general question, Carmen, you know, once you know the protein structure and the glycan decoration, you know, you're off to a very good start. But of course, that only helps if you have a crystal structure of the protein structure. With the Nobel Prize in Chemistry this year now being awarded to two of our friends in Google DeepMind, who now can more or less accurately predict the structure of any proteins once they know the amino acid sequence. Has any of that work impacted on the glycochemistry you've looked at where the crystal structure was not available?
Yes, quite a lot. Actually, we are now doing this using, when the crystal structure is not available, but an alpha fold, what we can predict it with alpha fold, for instance, this also works. Then we can dock the acceptor and the donor using docking, we also always need to check with experimental information, of course, with mutational information, whether that makes sense or not, and we can get a more reliable or less reliable initial model. But that's a very good, excellent alternative when there is no crystal structure. Exactly. We are doing this in some cases, yeah.
Yeah, super. Again, open for questions from the audience. I don't see any coming through in the Q&A.
Everything was clear.
Everything's clear.
Or maybe very
all
unclear.
Good, all good, all good. So perhaps just in terms of O'Magariel raised hand, Asa, okay. Let me just go to that. My colleague, who I think you know because you visited here very recently, attendees, oops, and where is Owen? Should be at the top. Is that right? That's right. So allowed to talk. So Owen, let's see if this technology works. You're only around the corner from my office, but let's see if it works.
Okay, so thank you very much. Can you hear me?
Yes, perfect.
Okay. Thank you very much for the talk, Karma. In one of the examples you showed the donor had been distorted into a 4H3 conformation. Can you say a little bit about how important is distortion and how much is the enzyme actively binding the sugar to distort it before the transition state gets to transition state?
Yeah, the one that I showed, this distortion was in the transitional state. Before the transitional state, in that case, there's no distortion. In the transitional state, the sugar is an oxocarbenium ion. It's practically an oxocarbenium ion because two bonds are being formed and broken. So the anomeric carbon is practically sp2. And then it gets planar around the anomeric carbon. So normally it adopts this type of half chair or envelope conformation. But before the reaction, normally they are not distorted, the sugar, in glycosyl transferases, which is what I'm focusing in this, what I focus here. Because the leaving group phosphate is already is axial, is in the alpha stereochemistry, and it's already very very well oriented for leaving, no, for acting as a leaving group. So the other one attacks from the top, and then it's quite easy. But in other carbohydrate active enzymes, like the glycosidases, the ones that catalyze cleavage of the glycosidic bond, not formation, in that case, the distortion is very relevant, because in that case, the sugars are distorted before the reaction, and they are distorted to place the leaving group axial to facilitate the reaction. However, in glycosyl transferases, I have not seen, and initially there should not be advantage in for the enzyme in distorting the sugar before the reaction, because the leaving group is already axial. Well, I cannot discard that there could be some exception, but it's not like the glycosidases, in which this is very important, because the glycosidic bond maybe is equatorial on all the beta glycosidases. It is equatorial at the start, so the sugar needs to distort to place the leaving group in a good orientation for catalysis.
Okay, thank you very much. That's very clear.
Great. Thanks, Owen. Thanks, Karma. So perhaps we'll go on to the next speaker. Again, thank you again. Over to you, Anton.
Yeah, thanks, Karma, for this outstanding and clear presentation. Yeah. So the next speaker comes from the Spanish National Research Council, FESIC, from the Institute of Advanced Chemistry of Catalonia in Barcelona. And Professor Pere Clapés will talk about engineered aldolases, the green lane to asymmetric carbon-carbon bond formation. Pere.
Okay, thank you very much. Thank you very much, Anton. Thank you very much, Pat, for inviting me to this interesting symposia. I was really honored to participate in that conference. And well, the topic I'm going to talk today is one of the lines that we have been working on for a long time, which is the aldolases, because we were interested in enzymes that can be useful in the carbon-carbon bond formation. So aldolases are a class of liases that, okay, this one, a class of liases that catalyze aldol reactions. They mainly are found in carbohydrate, amino acid, and hydroxy acid metabolism, mostly esteemed for metabolic degradatives carbon-carbon bond cleavage. So they are degradative enzymes. From the stereochemical point of view, what we have seen with many aldolases is that they are stereospecific for the approach of the electrophile to the nucleophile enzyme complex, which is the activated species with the donor species. And there is the fascial differentiation of the electrophile is the responsible for the relative stereochemistry. And in this point is that the electrophile is the one that makes more difficult to make the reactions stereoselective, although most of them are fully stereoselective. So from the activation point of view, they are classified in class 1, where the enamine is the active species, and then the class 2, which are always metallo-liases or metalloaldolases, in which the Lewis activation is done with the metal, normally zinc or cobalt or other metals. Then there will be some other that use cofactors like pyridoxal 5-phosphate, in which the quinonide complex is the donor species that reacts with the aldehyde, with the electrophile, normally are aldehydes. And then also the thiamine diphosphate. These enzymes catalyze, let's say, the benzoene reactions, okay, and acting at prolonged activation of the carbonyl, so to change the activation or let's say that what is it called, the electrophilicity of the donor acceptor substrate. Okay. For the aldolases also were classified in the past for the nucleophile specificity. Nucleophile specificity was classified as pyruvate aldolases, dihydrogen phosphate aldolases, acetaldehyde aldolases, and glycine aldolases. Okay. And normally these enzymes were quite flexible for the electrophile extraction, so they accepted many aldehydes. However, with improving in the molecular biology techniques, particularly directed evolution, particularly the structure guided approach, we can engineer aldolases to improve the selectivity towards the nucleophile, towards the electrophile. Even the most elusive thing maybe is to change the stereoselectivity of the aldolases. But in some cases, we have found that it is possible, but it's the most elusive way to change, or the most elusive parameter to change in the aldolases by a structured guided approach. So we have been working in our lab with many aldolases, starting with the HAP aldolases. Then we've also worked with D-fructose 6-phosphate aldolases, the DIRA aldolases, that is from the acetaldehyde dependent aldolases, benzaldehyde liases, which is typically a TDPH aldolase. Then 2-oxoacid aldolases that we classify because it's not only pyruvate the substrate, it can accept many other 2-oxoacid derivatives. Then L and D-threonine aldolases and L-serine hydroxymethyltransferase, which is acting very close to L and D-threonine aldolases. And for this talk, I have selected 2 aldolases, which is the D-fructose 6-phosphate aldolases and 2-oxoacid aldolases, particularly KPHMT from E. coli. And I selected these as model examples of how we have organized our work on aldolases. And this was in combination. Also, we use these aldolases in combination with others to make new compounds. And from that point of view, I thought that maybe it will be interesting to talk about these two aldolases. Okay. The fructose 6-phosphate aldolases, also called FSA, was discovered in 2001 by Professor Sprengen in Stuttgart. And it was found immediately that this aldolase was able to reversibly catalyze the cleavage of the fructose 6-phosphate into into dehydroxyacetone and diglyceraldehyde 3-phosphate. The physiological role of FSA is unknown, still is unknown. The natural substrate is unknown, so it was a quite rare enzyme in the E. coli. Well, the crystal structure shows decameric structure, okay, built up on 2 pentamers, packing face to face, and this so compact structure makes this enzyme extremely thermophilic, okay, keeping the activity after a treatment of 70 degrees for between 40 and 45 minutes. So the subunit of FSAI consists of a single domain that folds into alpha beta bar, which is in this slide. This is the single domain, the subunit of this decamer. Okay, in FSA we can this is the active site of FSA, and we can distinguish two sites: the nucleophile binding site, which is this area of the active site, and the electrophile binding site, with this will be this one which is located the glyceraldehyde 3-phosphate, and this nucleophile binding site is located DHA enamine. Okay. Then, and I would like to show you that by modifying these 6 amino acids, we have constructed a toolbox of FSA variants that can fulfill a lot of different aldol reactions. Well, from the very beginning, it was a really extraordinary enzyme, because it was, the main advantage of this FSA over the other DHAP-dependent aldolases is that it can use dihydroxyacetone. And dihydroxyacetone is not necessary to be phosphorylated, so this will, streamline the process of synthetic process, because it avoids many steps for the formation of the dihydroxyacetone phosphate, for the phosphate group. It was found at the very beginning that it was also very active with hydroxyacetone and hydroxybutanone, which, as I told you, it was very exceptional for at that time for the aldolases. With protein engineering, I will show that it can accept also small aldehydes, ketones, hydroxyglycolaldehydes, and also other alkanones and ether-derived alkanols derived with aliphatic alkyl chains, branched and linear. Okay, from the electrophilic tolerance, we have seen that we have observed that it accepted amino aldehydes, conveniently protected. It also accepted hydroxyaldehydes, phosphorylated aldehydes, of course, and aliphatic aldehydes like acetone, acetaldehyde, propanol, and so on. So the first example was how we can modify the substrate of the donor specificity. At the very beginning, we observed that the tolerance of the FSA wild type was hydroxyacetone, followed by glycolaldehyde. Already we have seen that a wild type accepted glycolaldehyde, and then much less dihydroxyacetone. And we were looking at the active site, and we saw that close to the nucleophile binding site, it was the alanine 129. Well, if we substitute the alanine 129 by a serine 129, it was this part of the nucleophile binding site, it will be more hydrophilic. So we hope that maybe we can change the sequence or the selectivity for the nucleophile, and that was this. With this modification, DHA was the best substrate, following the hydroxyacetone, and then at the end with the glycolaldehyde. And you can see here with the activity of the FSA, we see that wild type the activity was almost nothing, while with hydroxyacetone and glycolaldehyde was very high. And when we apply this mutation, the dihydroxyacetone becomes the best activity, or the best substrate for the FSA activity, followed with the hydroxyacetone and then with glycolaldehyde. Okay. Also, observing the nucleophile binding site, we thought that maybe, or we observed that two leucines, leucine 107 and leucine 163, there is some kind of wall that precludes to extend the donor substrate towards one of the ends of the, for instance, in the extreme of the hydroxyacetone or hydroxybutanone. So changing these two amino acids but alanines, substituting these amino acids but alanines, it will be possible to accommodate, long chain alkanones with linear ones, with linear chain, also with branching or fully branched chains, and also with ether-derived hydroxyalkanones with also derivatized with functionalized moieties. Then we apply these mutants, is a series of these variants, to the reaction of the aldol reaction of these donors with 3-hydroxypropanol, and then we obtained this a collection of deoxysugars, all modified in the anomeric carbon with all these functionalities, long chains, and also with this allelic function. Okay, next, we were very interested in the activity of FSA towards glycolaldehyde, because this activity will give us another aldehyde that can be, perform new aldol additions and produce multifunctional products or even molecular scaffolds. For this, we wanted to engineer again the A129 residue, which is connected to the selectivity, as we see before, the selectivity of the donor substrates, and with applying saturation mutagenesis at this position 129, we found that 3 clones that are really active against glycolaldehyde, which is A100, with the substituted with lysine, threonine, and valine. And we use for this the homoaldol addition of glycolaldehyde, giving the molecule of D-treose, okay? And we use this molecule to identify what is the clone most active with this substrate. Okay, after that we modify some residues in the electrophile binding site, like alanine 165 and serine 160C, and for instance, with the collection of the toolbox with these FSA variants, we will be able, for instance, to perform the stereoselective trimerization of glycolaldehyde and glycolaldehyde derivatives to, produce, for instance, D-idose when R1 is OH in a conversions between 58 to 99 to 98% and isolated yields to 11 to 48%. Also, when we investigate the S166 position, we were able to change the stereoselectivity of the FSA, and instead of giving D-treose, we were able to produce L-erythrose, which with another, addition of another equivalent of glycolaldehyde, we end up with L-glucose with a conversion of 81%. And with this molecule, which chemically it's quite difficult to synthesize. Okay, we go with other aldolases that I have shown you in the first slide. For instance, with benzaldehyde lyase, and we have a combination of these aldolates with the FSA variants to produce C-aryl carbohydrate derivatives. And the strategy in this case was very easy. It was, we don't remember that benzaldehyde lyase, it was a THDP dependent aldolate, so unpolum activation of the carbonyl groups. And this will react with the etoxyacetaldehyde to give this kind of compounds after reduction and unmask this aldehyde with treatment with acid. And then we applied the FSA variants, and with different variants to accept dihydroxyacetone, hydroxybutanone, or hydroxy acetone or glycolaldehyde. and we end up with different C-aryl derived fruit sugars like C-aryl sorbose, fructose, tagatose, and C-aryl L-xylose derivatives. So another application of this of FSA was in the synthesis of aminocyclitols, pipicholic acid, and pipicholic acid derivatives. So in this case, we use amino acids or non-coded amino acids to produce conveniently protected aminoaldehydes. And then applying the FSA variants, we can build up this kind of scaffolds that upon the protection, we can make different iminocyclitols or pipicholic acid derivatives. But we are not always, we want to expand the collection of amino acids and non-coded amino acids with other enzymes, which call it L and D threonine-aldolases. With these enzymes, we can construct beta-hydroxy amino acids, which we will transform into also alpha amino acid, alpha aminoaldehydes, beta-hydroxy aminoaldehydes, which also, applying also FF variants, we can also produce different scaffolds that can be converted in a variety of hydroxylated pipicholic acid derivative, glycosylated piperidins, pyrrolidins, pyrrolicidins, indolicidins, quinolicidins, and so on, with different degrees of hydroxylated compounds. And all these were tested as glycosylate inhibitors. Okay. Next, a question we ask ourselves is this the hydroxymethyl moiety is necessary for the activity of FSA, as donor, to accept it as donor? And for this, we have found some incipient activity of acetaldehyde in wild-type enzyme. So that means that we can modify the FSA to make this more active. So, and the residue we focused it in aspartic 6, which is connected with the nucleophile binding site and with the electrophile binding site. Okay, and we substituted this amino acid, but some hydrophobic amino acids, some basic amino acids, acid amino acids, but a little bit longer, some isosteric amino acid, but with only not acidic, asparagine, glutamine, and then serine and threonine. Okay, and to our surprise, none of these amino derivatives were active towards the, let's say, model reaction. Only the wild type and the isosteric analog asparagine were active with this reaction. But if we are not, of course, not interested in the reaction, we are interested in the reaction with other nucleophiles. And fortunately, we have found, or interestingly, we will find that these were active towards acetaldehyde, acetone, butanone, pentanone, cyclobutanone, and cyclopentanone towards the phosphorylated aldehydes, these 3-hydroxypropanol, and also other aminoaldehyde derivatives to produce, for instance, this kind of deoxysugars. Okay, and also these derive from phosphorylated beta-hydroxyaldehydes, and these derive with the 3-hydroxybutanol. Also, the ones less hydroxylated piperidine derivatives and were also produced from the conveniently protected aminoaldehydes. Okay, the second strategy we in collaboration also with Professor both did Fesnery in Darmstadt was, why don't we also target this amino acid, the threonine 26, because it's connected to aspartic 6 with hydrogen bonding, and also can affect as well the selectivity of the fructose 6-phosphate aldolase. Okay, we target these two residues with a small alphabet because we don't want to screen the whole 20 amino acids in each position. We wanted just to screen some very few amino acids, small amino acids. And with this small alphabet for each amino acid, we just have to screen 200 clones for a 95% coverage. I mean, 90% coverage means that 95% probability of screen all the amino acids that we have selected. So we selected for this reaction, we selected a screening assay. The screening assay consisting in a mixture of propanol plus acetone in the proportion of 1 and 17 equivalents of acetone. So very stressing screening assay. And what we found is that when we use D6H, for instance, FSA variant, we only obtain the cross aldol addition reaction, so the aldol addition of acetone to propanol. But when, for instance, combining D6H with T26L, we were able to exclusively form the homoaldol addition reaction, although there is a large excess of acetaldehyde in the screening assay. So that means that this enzyme is exclusively accepting propanol as donor and acceptor. Okay, the next enzyme I want to show you is the ketopentane hydroxymethyltransferase. And this is an enzyme that is in the pathway of the synthesis of coenzyme A and is responsible to the aldol addition of alpha-ketoisovalerate to formaldehyde. And the formaldehyde, of course, it cannot be freely in the cell because it's a highly toxic substance and is masked as N5-N10 methyl tetrahydrofolate. And this is producing the ketopentane, and the ketopentane follows then by ketopentane reductase to form the pantothenate, which is the vitamin B5. But it was discovered also these aldolates can be operate under the tetrahydrofolate independent mechanisms. So we can mix alpha-ketoisovalerate with formaldehyde to get exactly the same product as with the natural reaction. Okay, the KPHMT is also decameric extraction. In the natural metal is a magnesium coordinated by all these aspartic acids and glutamic acids. So all is acid residues. And what we found, we asked first is other metals are more active than magnesium, okay? So we can construct an artificial KPHMT. And answer is yes. And exploring this, let's say a natural reaction, we found that the best metal was cobalt, and then following by nickel, and then magnesium. Of course, with the DTA, the enzyme is completely inactivated, and with no metal added, some magnesium is still present, and there is some background reaction with after purification of the KPHMT of the enzyme. So in that case, we also propose to improve the nucleophile specificity. For that, we target all these residues, of course, that with the structure guide approach were found to be in contact with the most sensitive or the most bulky moieties of the donor. And with these modifications, we found that it will be useful to use them to produce beta-hydroxy acids using all these different donor substrates, okay? Some of these with functionalization, okay? But the second application that we found most interestingly was that the biocatalytic constructions of quaternary centers. So the quaternary centers are important for us because it can expand the chemical diversity besides the stereoaromatic flat chemistry to enhance the binding selectivity, avoiding unspecific interactions, and have better solubility and bioavailability. So with KPHMT also performs a reaction which is producing a quaternary center. So we thought that maybe we can improve or we can expand the nucleophile specificity toward all this plethora of 2-oxo aldehydes, and also combining with different electrophiles. So we found that with this methodology we can find, for instance, 2-oxolactones bearing geminal groups or even spiral groups, 3-hydroxy acids also bearing some quaternary centers, and ulosonic acid type products with that we are precursors or can be precursors of antiviral compounds. And thus, we started with mixtures, enantiomeric mixtures of these two chiral centers, and we end up with pure compounds or relatively pure decent enantiomeric ratios. And well, with this, I will finish. If I have some time, I would like to explain. I would like to tell you some of new research that we are going on. And the first one is the construction of artificial metallolysis, that is installing artificial metal cofactors into bioscaffolds. And one of these, the starting was allotac, which is allotac with one mutation is not able to perform internally the hydrolysis of this bone, and then we can make it, perfectly well or very selective to anchor the ligand to this scaffold. And here we wanted to also to install a scaffold steering pyridoxazoline or bisoxazoline or bisazoxazoline to make artificial metalloenzymes. Also, another enzyme that we have recently discovered was the DDK. The DDK is performing the cleavage of dimethylsulfonate propionate into acrylate and dimethyl sulfide. And we thought that since this is a beta elimination, if the enzyme can work on the other way around to make the aldol or Michael additions to acrylate and acrylate derivative. And actually we started with Asa-Michael reactions in which the enzyme has a very broad selectivity with different primary and secondary amines. And also we found that it was also active towards methacrylate, and the product of methacrylate was enantiopur, the one obtained with enzyme. So we think that in this field we can build up a new enzymes to perform new carbon-carbon bond formations and new carbon-nitrogen bond formations. And yes, and just to finish, I would like to thank my students, the staff, scientific staff that we are in our group, the postdoc, the current PhD students, and the former PhD student and postdoc. Also, the external collaborations with different groups, and also the financial support was always very necessary, to perform the research. And with this, I would like to thank you for your attention. Thank you very much.
Great talk, Perry. Again, 4 in a row. Great talks, and this is all to do with engineering aldolases. And again, the paper is open for questions from either the panelists. 2 participants have raised their hands. So Oscar is first up as far as I can see. Oscar, you can...
Okay, very nice talk. I have a question because it was not clear for me. When you choose the residues to make the modifications, have you done any calculations, or it was more intuition, which is sometimes is better than...
It's a mixture of intuition and calculation, because what I first do is to observe the molecule myself, and more or less, by intuition, see the amino acids that I could change according to the size of the molecule, and then I send my guess to the computational people, and they say, well, you are right or you are wrong, and well, you have to choose another residues, or you can choose your strategy is completely wrong. No, but normally the intuition it gives you a lot of good signs, you know, because it was sometimes they contradict me, but many times are agree with me. Yes.
Okay, thanks.
Thanks, Oscar. Anton, over to you.
Yeah, great talk, Pere. Thank you very much. I have a sort of philosophical question, which are always very difficult to answer. So now the synthetic community has more or less three platforms, no? The standard transition metal-based catalyst, organocatalysts, and enzymes or engineered enzymes for the chemistry you have shown. What's in your opinion the future? Will any of these three platforms displace the other two? Will the three of them always coexist? And apologies for the difficulty of the question.
No, it's actually a very interesting question because I also ask myself this question, and I think that we have to. There is all these platforms are okay, and all these platforms are useful, and I think that not any platform will displace the other one. So all platforms are necessary. And I think that the future will be some complementary, platforms. I mean, we can complement biocatalysts with metal catalysts, for instance. We can complement organocatalysts with biocatalysts. And as I have seen publications for Professor Melchiorre, which is also showing that it is possible to combine organocatalysis with biocatalysis. So, in my opinion, is a mixture of, or it will be, a combination of all these techniques to obtain the molecules that we want, because biology is demanding many difficult molecules to chemists. So we have to have all the tools necessary in hand to solve the problem. So it is as many possibilities as you have on hand, you can solve the problem easily. I mean, there is no just one way to do the things. Fortunately, we have many ways to attack the problem, and this is one of the greatest things of the chemistry, that there is many ways to attack the problem, and then we can solve many problems so far. So we are not at the same level at 20 years before, and we expect that next 20 years the chemistry will evolve in a position that they can solve most of the problems that biology are putting to us. And I should say, for instance, this field of artificial metal-aligned enzymes is, I think that is very interesting, because once there is activity, or there is enhanced activity with the artificial metal enzymes, the enzyme or the bioscaffold can be evolved. The ligand cannot be evolved, can be made many chemically many ligands, but we cannot not evolve biologically and ligand. So from that point of view, I think that will be very exciting field in this field between metal chemistry and biological chemistry.
Good philosophical question, Anton, and many people around the world are thinking about what is the right answer to that. Perry, good answer, well done. Perhaps in the interest of time, we might keep moving on, if that's all right. So if I could ask you to maybe unshare and leave it over to Anton to introduce Oscar, who's our next speaker.
Next speaker comes this time from the University Rovira i Virgili, south from Barcelona, in Tarragona, and he works actually in the physical and inorganic chemistry department of this university, and the title of his talk is Expanding the Scope of Asymmetric Hydrogenation to Include Tetrasubstituted Enones. Oscar, thanks for accepting the invitation, and the audience is yours.
Okay, first of all, I would like to check can you see that something is moving in your screen?
No, I'm afraid.
Okay, then
it's a static, which is always wrong.
Yeah, then I will go to another option, sorry, which is the PowerPoint presentation, so hopefully it will work. Now is moving something?
Yeah, now you just moved to research group interests. Yeah.
Okay, great. Thank you. Thank you very much. So first of all, thanks for the introduction, Anton. But first of all, I would like to thank the organizers, Anton and Pat, for the opportunity to present some of our work here in the community. It's also a pleasure to see how people is doing around. Okay, so our research group interest is the design of highly active and selective catalysts for reaction of industrial interest. And to achieve this goal, we make 3 different approaches. So the first one is the design of modular ligand libraries. Then the use of continuous flow with immobilized catalysts. Usually these modular ligands libraries, we immobilize them. And finally, the use of machine learning and predictive transition stage force fields for the development of the modular ligand libraries. But today I'm going to talk about asymmetric hydrogenation. And this is a old reaction that has become one of the most powerful methods for the production of enantiomerically pure compounds, mainly because of the it is a reaction that is very easy to perform and it's 100% economical, so industry really like them. Okay, from that we can classify asymmetric hydrogenation depending on the type of the olefins that we can use. So if we use chelating olefins, so olefins that coordinates as a beta tail to the methyl center, then like the hydroamino acids, then this has been highly studied, and rhodium and ruthenium diphosphine catalysts are the state of the art. If we move away from this and we go for non-chelating olefins, then this is a much underdeveloped field. And in this case, iridium PN catalysts are the state of the art. To make a brief history of this part, okay, I would say that this came from the pioneering work of Fals when he discovered that iridium PHOX ligands, iridium PHOX catalysts were exceptionally very good for the asymmetric hydrogenation of trans-methyl stilbenes. Okay, so since then, the ligand has been modified by replacing the oxazolene by other nitrogen or sulfur donor groups, and the phosphine by other P-related ligand, P-related donor groups like phosphinite and carbene. And of course, the ligand scaffold has also been changed. But we can say that this led to the development of a series of phosphinite nitrogen ligands that work very well for tri-substituted olefins. And here you can see some of the 3 more characteristics for this transformation. Our group early demonstrated that the introduction of a biaryl phosphide moiety into the ligand design, it was beneficial in terms of substrate scope, because then we can not only hydrogenate tri-substituted, but also di-substituted olefins with excellent anti-selectivities. However, tetra-substituted olefins were still a problem and we cannot hydrogenate them. In order to know why, we decided to make a computational study, and the results indicate that the mechanism of our catalyst follow the one that has been already predicted by many others, that is the iridium-3, iridium-5 migratory insertion catalyst, in which the transition state for the migratory insertion is the key for the anti-selectivity. By studying these transition states, we discover a quadrant model. Okay, what is going on? Sorry. We discover a quadrant model in which we can see that our ligand blocks the upper left quadrant, making totally hindered. So there is only here, we can only have a hydrogen on it. And in the lower right quadrant is semi-hindered. Okay, so leaving the 2 other quadrants open, which is really good for E olefins, which has large trans substituents. But the beauty of the system is that the biaryl phosphide moiety is flexible, so it can accommodate Z olefins by tuning the occupancy of the lower right quadrant, as well as for di-substituted olefins. And of course, this model explains why we cannot have any result. We don't have any good result for tetra-substituted olefins. Okay, because here we can, in the hindered quadrant, we cannot put any substituent. So the asymmetric hydrogenation of tetra-substituted olefins is really a challenging task. There has been some reports on it. So for instance, Bagbal in the early 90s discovered that this zirconizine catalyst was able to hydrogenate some tetrasubstituted olefins. However, it was really low. The stability was really low, so we require high catalyst loadings, and therefore is impractical. But the real breakthrough came again when Falth and coworkers discovered that by modifying the ligand scaffold and introducing a 5-member clade ring, then you increase the room, so you open the chiral pocket, you make the chiral pocket more open, so the tetrasubstituted olefins are able to coordinate. And this paved the way because these catalysts are highly stable and you don't need high catalyst loadings or high pressures in order to perform the hydrogenation. And this has been led by others like Fer Anderson and many other, well, not so many, and few others in the hydrogenation of tetrasubstituted olefins. And the last example that I want to highlight in the literature is this rhodium diphosphine catalyst developed by Zhang, in which the reaction performed well, but again, long reaction times and high catalyst loadings are required. And in this case, I think that the most painful part is the ligand synthesis, okay, that is synthesized in 9 steps. So another important point that I want you to remember is that in contrast to other catalytic reactions, the asymmetric hydrogenation of tetrasubstituted olefins is highly limited in substrate scope. And small changes in the nature of the substituents, like for instance, changing a methyl by an ethyl, that usually does not change so much on the EE of the resulting reaction, here it changed a lot. So this small change can cause a detrimental effect on an anti-selectivity. So you can end up from, I don't know, from the methyl, you can have 95% E. And if you change methyl by ethyl, then you can end up with 60% E. Okay, so really huge effect on the substituents. Our group, in collaboration with Professors Riera and Verdaguer, developed some catalysts for this. And we have seen that we could hydrogenate some type of olefins, tetrasubstituted olefins, but what we were interested in is the asymmetry, if we can hydrogenate alpha beta unsaturated enons, and more especially the linear ones, because they are more difficult than the cyclic ones, okay, because you have more freedom. Okay, why is this? Because we can generate 2 bisinal stereogenic centers to the keto group, which is very good for post-functionalization, and because this type of skeletons is present in many natural products as well as drugs or intermediates for building that. There is one present in the literature when we started, and is this paper here from the Lilly, that they use the rhodium Josephus system for the asymmetric hydrogenation of this enon. Okay, and they end up with an anti-selectivity from 77 to 96% E. Okay, so really depends on the nature of the substituents. We then decided to tackle this problem. We decided to test this model substrate with this substrate here in the top. And we tried first the Lilly system, but the system was not good. Okay, the rhodium catalyst in our hands for this model substrate only provides low conversions and very low anti-selectivities. So we decided to move to iridium catalyst. In the first round, naively, we can say that we used the ones that were good for tri-substituted enons. And as you can see, that doesn't lead to any conversion. And then we moved to the ones that we know that work well for tetrasubstituted enons, for tetrasubstituted olefins. And the only good result that we can manage was this one. And this was our best result with the false ligand, but only had 11% conversion and very lousy in anti-selectivity. But this encouraged us to take this design and to replace the CH2 by an NH. And by doing this, we increase the conversion and then anti-selectivity. And since the synthesis is very easy of this new iridium catalyst, because the synthesis is shown here, and it can be performed in just 3 steps with just 1 column chromatography at the end. So it's very easy to build up a small molecule, a small library of iridium catalysts. That is what we did. And we found that the ligands that contain terboethyl groups at the oxazoline position were the ones that provided higher activities and higher diastereoanion antiselectivities. Here you can have some examples. Okay, and then we decided to lower the pressure. Okay, at 50 bars, we still have the same diastereoselectivity and E, but then we moved to 1 bar. And at 1 bar, the catalyst is still active, is still an antiselective, but the diastereoselectivity decreased. In order to understand what was going on, we decided to make some deuterogenation experiments. Okay, so we can see that at high pressure of deuterium, the deuterium is only incorporated at the correct carbon, so at the olefinic carbons. However, if we do the same experiment at 1 bar, we can see that there is a lot of deuterium incorporation at the methyl, at the alpha position of the keto group, which clearly indicates that there is an isomerization. And this isomerization is the one responsible for the deleterious effect on diastereoselectivity. Because now the deuterium can, when this terminal olefin is hydrogenated, the deuterium can enter to both sides of the olefin. So with the standard conditions, we can say we made the catalyst screening. And as you can see, the catalyst is quite robust. It allows several decorations at the aryl group. Okay, it also allows changing the geometry from E to Z. Okay, with the example here. Okay, regarding the functional group tolerance, we can see that it's quite good because it tolerates boron, boronic bipene, esters, Wenrepamides, and so on. And also it tolerates well modifications at the other substituent at the beta position. Different substituents at the close to the carbonyl group, and different substituents at the alpha and carbonylic substitutions with these cyclic enons. Okay, so the system is very stable and it's very robust and can work perfectly well for several substrates. So with that, we decided to perform some derivatization studies. And here we were a little bit worried because, as you know, we have a chiral center close to in the alpha position of the keto group. So we thought that we will have problems of epimerization. Okay, but this was not the case. So we can perform, for instance, the Suzuki coupling, hydroxylamine formation, Wittig olefination, and Grignard reaction. And even though these reactions are done in basic media, I mean that no epimerization was observed. And also we were able to selectively reduce the carbonyl group. So, and this is important because by doing this, we can form these 1,2,3-methyl OH stereotriates, which are present in many natural products. And it's a way to do that by asymmetric hydrogenation. And then, of course, we wanted to know what was going on. So for that purpose, we make a DFT study. And then we found that the most stable iridium hydride species is the one that is shown here, in which we have the enon coordinate as a bidentate. And where we can see that the double bond is coordinate trans to the hydride and the oxygen is coordinate trans to the phosphorus. However, from that, all attempts to perform in silico, of course, the 1,2 insertion were not possible. They were so high, they were very high in activation energy, so the transition states were very high in energy. So from this, the system has to evolve and the system has to isomerize in order that the enon is now, the oxygen of the enon is now trans to the hydride, and then the double bond can be trans to nitrogen or trans to phosphorus. Okay, and this goes the 1,2 insertion with a small barrier of 11 kilocalories per mole, which is feasible under the reaction conditions. And from this compound that has the vacants here, the typical thing would be that this will be covered by hydrogen, by coordination of hydrogen. But then here we observe that the coordination of hydrogen has a higher barrier than the 1,2 insertion. Okay, and that is weird, we can say like that, and that we found it a little bit weird. And so we said, okay, maybe this is not the pathway. So the reaction will not go via iridium-1, iridium-3 catalytic system. It will go via the typical iridium-3, iridium-5, in which the substrate is only coordinated through the double bond. We studied that, and what we found is that the barrier was even higher. So it is clear that the reaction does not go to iridium-3, iridium-5. So then we move back to this intermediate, to the intermediate 3. And then we found that in order for the reaction to proceed, what it has to happen again is a rearrangement, so an isomerization of the complex, in order to place the vacants trans to the hydride. And what is very peculiar of this system is that when we have this disposition, the reductive elimination is much faster, or is preferred. The reductive elimination is preferred than the coordination of hydrogen. So the reductive elimination happens first than the addition of the new hydrogen atom, hydrogen molecule. And this is the scheme of the energy profile of the reaction. And as you can see, the key intermediate is the first intermediate. So the 1,2 insertion, the intermediate for the 1,2 insertion, which is rate and an anti-selective determining step. Okay, so in order to prove that, in order to confirm that, we decided to calculate all transition states, all possible transition states for 12 different enons that we have previously shown that it provides good anti-selectivities. And we can see that the calculated is and experimentally is quite very high in agreement, which makes us confident that this is the key intermediate for the anti-discriminating step. In order to understand what was going on, we decided to study all the transition states. But here we have 4 transition states because the olefin can coordinate trans to phosphorus and trans to nitrogen. Okay, and here we can see that the ligand imposes a different aesthetic environment over the quadrants. So if you remember before, most of the ligands, our ligands and most of the others, has a trans disposition of the aesthetic environment. So we have a highly hindered one quadrant, and in diagonal position, we have the semi-hindered one. In this case, it's different. Okay, all are in the same site. Okay, leaving the other 2 sites completely empty. Okay, that's a different. This is a key difference between one and the other. Okay, and here you can see the scheme or easy scheme of the transition states, the 4 transition states that we can have at the bottom. And as you can see, in all these transition states, the beta substituents are allocated in the completely free quadrants. Okay, which indicates that we can actually, the aesthetic hindrance here can be whatever we want, and in fact, this is what we observe in our catalytic results. Okay, this also indicates that the substituent at the carbonyl group, okay, can have an effect if it's really hindered, but if not, it shouldn't be a problem to tolerate this. Okay. However, the methyl substituent, the substituent in the alpha position, this can cause some problems, and in fact, this is what we observe. Okay, so this DFT study allows us these model quadrants, and from these model quadrants, we can clearly explain our results. And as Arjan said, it's very important to understand why this happens. Okay, so we were very happy for that. Okay, and we are convinced that we have a very good catalytic system. So this is all. Okay. I hope that I have convinced you that we have been able to prepare a novel family of iridium catalyst precursors that are highly modular, they're very easy to synthesize in just only 3 steps, that allows the fast hydrogenation of linear enons with excellent diastereo and enantiomer selectivities. Finally, I would like to thank the people that has been involved in this group and this work, all highlighted in red here. The boss of the group, Montserrat Dieguez, and all the financial support, and last but not least, you for your kind attention.
Oscar, super stuff as ever, and oxazoline's close to my heart, so I enjoyed that talk. So asymmetric hydrogenation, perhaps your new title for this talk should be Old Reaction, New Tricks. So excellent.
Thank you.
So there's some 2 hands already up from panelists. Perry is number 1, and then Ariane. So Perry, over to you first. Perry?
Thank you very much, Oscar, for your nice talk. And I would like to ask you the same similar question of what you asked me before. What you use to design the metal ligand? I mean, no, I mean these quadrants sounds very useful to me, but I don't know how you figure it out. How is the stereochemistry and in connection with the ligand design, and what kind of rationale do you use?
Okay, yeah, I understand your question. And for this, there was no design. Okay, we usually, when we have when we know the substrates or we know another reaction, and we are pretty sure about the mechanism of the reaction, then what we usually do is we do some in silico study, prioristic study. Okay, but here there are no results. So we didn't know anything about the transition states. We didn't know anything about this. So what we did is just try an error, we can say. Okay, first we start with the rhodium that worked for the cyclic. We see that it didn't work, and then we went through all the iridium catalysts that we have. Okay, and then we end up that actually one that is not in-house design was the best one. But and then what here, yes, the change from the CH2 from the nitrogen, that was intuition, because we have seen that in many other cases the use of, for instance, any N-phosphines like this one or phosphoramidites usually provides better activities than the use of phosphines or phosphites. So that was more rational, we can say like that, but actually this was just empirical.
Okay, yes, but because I noticed that you always use phosphines, okay? Or
yeah.
Okay. It is necessary, this group, or you can design something without phosphines?
Phosphines you can use, I mean, all P donor groups you can use. We really like phosphites, okay? Even though this is not the case, because they are very adaptable, even because the BRE moiety is flexible. It seems rigid, but it's flexible, so it allows you to accommodate the different substrates here. So different aesthetically encumbered substrates. So we like it a lot, but at the end is when you have a problem and you want to solve the problem, you use all the tricks you can do in order to do that.
Okay. Thank you very much, Oscar, and I look forward to meet you anytime in Barcelona.
Okay. Thank you. We will do that.
Yeah.
Right. Over to you, Ariane.
Okay, so thank you, Oscar, for the great talk and congratulations with the results. I think that these are really amazing results in asymmetric hydrogenation. I was wondering about this new ligand that you designed, so this aminophosphine essentially. And I was just wondering about the potential non-innocent behavior of that NH fragment. So would it be possible, mechanistically, that that is involved in the orientation of the enone? Because it could form a hydrogen bond with the keto group and orientate the conformation such that you get productive catalysis. So did you look into that? That is basically my question.
Yeah, we have looked into that, and the first answer is no. It doesn't play any role here. Unfortunately, otherwise it would have been very, very nice. Okay. This work took us more than 2.5 years, we can say like that. And 2 years was just to discover what the heck was going on with the mechanism.
Okay.
So we explore all possibilities that you can imagine.
Okay. No, it's good to know, because potentially it could be involved, of course. But and just a short follow-up question. When you talked about the potential epimerization of your products when you do the post-synthetic modifications, so this must be pH dependent, right? So depending on the base that you have, there or not, depending on the post-synthetic, let's say, conversion that you are investigating. But did you try maybe to deliberately, epimerize, so with a very strong base, LDA or something like that? I mean, is it at all possible?
We have not tried that. Okay. And now I see some of your point. Okay, now I think that this is very interesting, because that will open up possibilities for DKR in the hydrogenation of the keto group.
Yeah,
okay.
Definitely.
Thank you for the tip.
Thank you for the talk.
Great, thanks, Arjen. And we have a final question then from Xavier Verdiger. Sorry if I pronounced that incorrectly. Verdiger, we can hear you.
Yes, yeah. Okay, thank you, Oscar, for this nice talk. These are amazing results. And I would like to insist on the nitrogen thing here. And so you said that there is a big difference between the carbon when you have the false catalyst with the carbon and the nitrogen. And you said that you kind of were expecting a boost in activity from changing from the carbon to nitrogen. And I would like to ask you what's the rationale behind that? Do you think that the aminophosphines are more electron rich or more electron poor than the carbon ones? Or so is that an electronic effect, or what's going on there?
Okay, I think that is an electronic effect, and this is an electronic effect. So I think that in this case, the NH, and in some other cases, for instance, Per Anderson has also found this. Okay, so this is not something that we only found ourselves. So Per Anderson has found this, and is the correct electronic behavior, the correct electronics for boosting the activity. Sometimes it works, sometimes it doesn't, because having the NH there, sometimes it makes your catalyst less stable.
Yeah.
Okay, so it's a balance. Okay, because if your catalyst then is less stable, then even though it's faster, then you end up with low conversions. Okay, so it's really a balance. So sometimes this trick works, sometimes it doesn't work. I don't have a proper answer for you, Javi.
Do you think? But let me ask you another difficult question. The aminophosphine is more electron rich than the carbon analog, or it's a less?
The M-phosphine for me, it's between the phosphine, we can say, with the carbon analog, and the phosphinite with the only one oxygen. Okay, so the donor, the basicity of this group is between these two.
Okay.
Where to place it exactly, I cannot tell you. We have not calculated any frequency, for instance.
Okay, so shall we leave it there if that's all right? Thanks very much for your question.
Thank you very much.
Okay, so Oscar, we might pause there. Thanks very much, and we'll pass over to Anton, who's going to introduce our penultimate speaker.
Yes, we should move on. We are a little bit behind schedule as we started a little bit late. So, next speaker is Professor Ana Belén Cuenca from the Organic and Pharmaceutical Chemistry Department from the Institut Químic de Sarrià from the University Ramon Llull, a long name that brings me very nice recollections. The title of the talk is New Reactions and Structures Involving Main Group Elements. Anna, thanks for accepting the invitation, and the audience is yours.
Thank you. Thank you very much. Number one, is the presentation showing correctly?
Yep.
All right, thank you. That was the whole point. Now everything will be good. So thanks a lot for the invitation. I'm very happy to be here to share you a little bit the chemistry that we are doing in our group. And then is behind this title, there is a group of people, talented chemists, starting by my close collaborator and co-PI of every project that we produce, that is Dr. Alexander Shafir, in the working in the EuChemS, and some of the already known in this audience institution, because some of the speakers are also coming from this institution as well. So then we are working in different areas. So basically, one of the main areas that we are working is with the hypervalent halogens, especially with hypervalent iodine. And then we work with them either from the reactivity point of view or with the structural point of view. We are also working in the group using the development for methodologies and structural new compounds containing boron nitrogen units. But today I will focus more into the CH reactivity developed with some of these hypervalent iodines. I'm sure that most of the people in the audience is familiarized or known or has been used hypervalent iodine chemistry in somehow, maybe probably for oxidation reactions. But just in case, I'm just going to go briefly until the description of this type of species. And then if we consider, for example, iodoarine here, and then you put the iodobenzene, for example, in the presence of an oxidant and in an acid media, you can end up sharing part of the electrons from the iodine, and the iodine is getting oxidized and increasing its coordination number. And then you can actually end up in a species that expanded the octet rule, and this is what we're calling the hypervalent iodine. Having here in the slide one of the most well-known species of this family, and because of his balance in stability and reactivity, that is the phenyl iododiacetate. That is the PIRA that we name it in the community that use hypervalent iodines. One of the interesting structural characteristics of these compounds is this T shape. So when the iodine oxidizes, these 10 electrons around it is forcing, or it is moving this species to adopt a curious geometry that is we know that on the basis of the X-rays, for example, here is the X-ray of the phenyliodonium diacetate, where you can observe that the two acetates are more or less 90 degrees with the carbon iodine bound, right? There is also some interactions between the carbonyl oxygens into the antibonding area of the carbon iodine bond. So these species their chemistry is highly governed by their tendency to get back to the reduced state. And this is probably one of the main well-recognized uses. Here, for example, we have an organic molecule, an amide, that when it interacts with the hypervalent iodine species, it is evolving, it is getting induced, it is participated in a Hoffman rearrangement with the aim of trying to iodine get to be reduced, right? So then also you can imagine a possibility of transforming these hypervalent iodine species into other hypervalent iodine species that are relatively stable, like, for example, diaryl iodonium species or other reagents of hypervalent iodine that are bounded to relatively electronegative substrate like CF3 or alkynyl groups, and those groups could be transferred to nucleophiles because of this high tendency of this carbon, for example, in this diaryl iodonium sol, and I here showing an example of isotopic fluorination using a bad nucleophile, which is the fluorine atom, but still possible to attack to this carbon and generate the fluorinated version, giving back the hypervalent iodine, the iodobenzene as a side product. So this, you can also find this species participating, this new stable hypervalent species in reactivity that is not essentially giving back the reduction of the iodine. For example, you have here a version of cyclic diaryl iodonium species, here monomeric, or here having a two iodines into the same molecule that can engage in molecular recognition phenomena, for example, or even they can be used in biochemistry, for example, as suppressors for NOx oxidase activity, or even recently has been discovered that this species, specifically this diaryl iodonium chloride, that it can act as an agonist of a GPCR receptor, right? So then today, within this context, I would like to tell you a little story that we have been working for a while, for some years, and it's a particular reaction where these phenyl iodo or aryl iodo diacetates are interacting with quite of saccharide type nucleophiles, silyl-based nucleophiles in the presence of a Lewis acid, and then in this reactivity, the iodine, the iodoarine is not anymore byproduct in the reaction. Rather, the two substrates are combined in a very regio-specific manner, and then giving essentially the formation of the carbon-carbon bond between the carbon bounded to the silicon and we will see if this is carbon, right? But then the carbon binds to the silicon, the propagylation of this ortho position. This is, of course, a reaction that has been described before us. So it's actually results from the group of OTI in 1991, and it has been revisited by our group in 2018. So then if we try to understand a little bit what it could be going on here, well, one can imagine that the first initial interaction between the Lewis acid, that by the way is essential for this transformation, with one of the two acetase groups in the hypervalent iodine reagent. And this is, we were lucky back in the years to be able to even characterize this first initial adduct by X-ray, and which you can see how one of these iodine oxygen bonds in the acetates is being clearly labilized and is opening the door for the incorporation of the nucleophile, right? If we can imagine an extreme version of this activation, we can even imagine the dissociation of one of these two acetates, and then this is opening the door for the approaching of the nucleophile, most probably by the multiple bond, right? So this interaction is nothing else than a saccharide paradigm reactivity, where after the coordination, the decylilative transmetallation, we say, let me use this word for all the presentation, trying to mimic in somehow making an analogy between the iodine and whatever transition metal, right? It will be, for example, a little bit of an EuChemS paradigm as well. So then upon this coordination, the decylilative transmetallation can take in place, generating this quite of alanine iodonium intermediate. And this is where the reactivity opens a possibility where, and under another classic paradigm for organic chemistry, which is the Claisen paradigm, you can imagine an evolution of a 3,3 sigma tropic reactivity in this alanine iodonium that will create a new carbon-carbon bond, thus obtaining this Wheelan intermediate that after elimination of the hydrogen will rearomatize and giving you the final product, right? So then this is nothing else that a formal ortho-CH functionalization of iodoarine guided by an iodine center, right? So then we were happy to obtain those kind of propagulated compounds, expanding and revisiting this kind of initially paradigm described by OTI. And then at some point we were wondering, what about if just trying to develop a parallel reaction instead of using propergyl silane, using allyl silanes, given that the allyl group is propergyl as well as, but the allyl group we consider it a very versatile functional group. So again, this reaction was a little bit described in the literature, and this is work by Zhu and co-workers. And then this reaction, as you can see in the scheme, it could be conceived under the very same paradigm, which the Lewis acid again will probably activate the iodine, and it will open the door for in this case, the allyl iodonium intermediate to evolve to the sigmatropic rearrangement, right? But the thing is that this reaction, which is very nice, is only working. It has a severe limitation in the implementation because it's actually needed a very highly electron excited arranged iodines to take place. And you can see here that the reaction is very efficient with just pure allyl silanes when you're using an iodine that it has a methoxy group, but when you just go for more neutral iodines, the reaction is really sharing low, and it's even almost impossible if you have electron withdrawing groups into the iodine. And then one of the possible rationales for this situation, it could be that if one imagine the allyl iodonium intermediate in this reaction, of course, one of the possibilities is that you evolve through the desired, let's say, sigmatropic rearrangement, but you can also imagine a possible decoordination in order to reduce for this high tendency that I told you of the hypervalent halogens to go back to the reduced state, and then you eventually will generate an allyl cation that it could be. In fact, these are detected product in the reaction media. It could be tri-body adventitious nucleophiles that you have in the reaction media. You can even see here a Ritter reactivity and O-acetate from the iodine, or even a clearly saccharide nucleophile, given the dimerization of the allyl silane. Of course, this process could be seen as an SN2 mechanism, but it can also be envisaged as an SN1, or it could be envisaged as an SN2 or SN2 prime mechanism, depending if the nucleophile attacks in the cis antibonding pi antibonding of this allyl iodonium intermediate. Also, of course, allyl silanes, nucleophilic silanes are electron-rich species, right, in general, and iodines are oxidating reagents, right? So then we can also one can also imagine a possibility of an unproductive unspecific oxidation of the nucleophile, right? Of course, a process that it will be less favored if you use an electron-rich iodine, right? So then at this point, we were wondering if we could be coming into the area, applying some of our knowledge from the proper dilation reaction, and also trying to reinforce to make a more robust allylation, CH allylation reaction. Yes, and we judge that the incorporation of an electron withdrawing group into the nucleophilic allyl silane might be beneficial, right? Because it will be, in principle, discourage or destabilize the cation, the possible cation, allyl cation, but also it could maybe stabilize the allyl silane nucleophile for the possible oxidation, right? In fact, this is some data. I'm sorry that I don't have the phosphonate example. It's just the ester here, but it could give us, it can give us an idea of how much stable are these allyl silane derivatives towards the possible oxidation, right? Then this is a work, by the way, with one of our beloved talented chemists, Wei Wen Chen, that it just defended his PhD. So then, of course, when we prepare these compounds by olefin, but ruthenium catalyzed olefin metathesis, and this reaction is working nicely. And then we tried the reaction, and then to begin with, we decided to try the relatively deficient iodine, because as I saw you, it was one of the limitations that the group of Zhu found while using just pure allyl silane. And turned out, as you can see here, that the incorporation of this electron withdrawing group group, it has a highly beneficial effect into the reaction, and that's just optimize the conditions. We just optimize the condition and figure that two equivalents of the Lewis acid are the optimal stoichiometry for this reaction to take place. This reaction was working in Wei's hands fantastically. And then we were able to incorporate many different iodines, many different as well functional groups here, electron withdrawing functional groups here, to perform almost 50 new molecules we were able to prepare with this methodology. You can see here some of the selected examples. You can do the reaction with the sulfone, with allyl silane having here an aryl sulfone group, even combining it with an iodine that has a clearly deficient nature with two chlorine atoms. This is also possible to apply to heterocycles. It is possible to do selective, of course, reactivity with this naphthalene. And then we also have tertiary amides, Wainwright amides, carboxylicases, acids, proline derivatives, and so on. So then we were actually happy because it looks like the incorporation of this group really opened up the reactivity for this alylation reaction. Then I didn't mention, but in all cases, the reselectivity is almost complete or super high, right? So then this reaction really has the tendency to go to the ortho position. So then, you know that the methodological people, we always need to kind of show that what we are doing is serving for something. So we decided to make some chemistry, some elaboration chemistry with this, taking the advantage of the ortho disposition of the iodine, that it gets reduced but it stays in the molecule in the final product, and the new allylated fragment. In this way, we were preparing this kind of vinyl diazo compound that it could be prepared one pod after the CH allylation just by simple treatment of the crude with the tosyl acid and DBU. So then this vinyl diazo could be engaged in just classical chemistry that I'm not gonna just go into detail, but in route to prepare this antimitotic agent, the dosabolin, in a much efficient way that it was comparing to the described synthesis. Just as for the sake of illustration, here we have a beautiful aromatic expansion that it could be developed upon the this particular CH allylated product using the beautiful chemistry described by Laroche, using benzynes arines as a insertive component in this reaction, and this is giving this kind of intermediate that it can be elaborated, and we elaborated towards the crisnatol, which is an intercalated anti-cancer drug. So then at that point, we were happy with the allylation, but we were thinking, what about if we are able to what about if we introduce a new insaturation into these systems? What about if we expand the conjugation system, the conjugated system in this silent nucleophiles, right? Could they engage and go to a reactivity in other further positions into the arene? So then the question will be if this allyl silane, standard allyl silane, behaves indeed as a three-component, giving rise the allyl iodonium intermediate that will eventually evolve towards the ortho-CH coupling, or if, on the contrary, the whole conjugated system could be involved in this reactivity, and then we may arrive to the par position or other position rather than the ortho in this system, right? Given what it could be seen as a pentadienyl iodonium intermediate, right? So the answer is that we were trying to prepare these compounds by olefin metathesis. It was not the answer. This is the synthesis. Sorry. So we were trying to prepare this by olefin metathesis. We succeeded, but it was a mixture of isomers. So it turned out that we needed to develop some cross-coupling chemistry even to prepare the compounds, but we succeed. This is a work with Ursula and Ines Edo. And when we test this reaction with just phenyl iododiacetate in the presence of triphenyl, in the presence of trifluoroboron as a Lewis acid activator, turned out that the reaction took place, and although it was not super efficient, the compound, the nucleophile was the CH functionalization took place into in a highly regioselective manner into the para position, right? So we were very happy to see this. We were doing some DFT calculation in the group. And then we did some collaboration in the calculation of this system with Miquel Solà, the computational chemist in Girona. And then we were finding that the interaction between the activated iodine and this molecule seems to be oriented in this way that I'm drawing in the slide, and the decellulation evolved directly to some sort of pi stack intermediate that is already quite highly oriented to evolve to the final reactivity. And here you can see that if one take a look to this pi intermediate and observe the orbital interaction, there is a perfect 3-point match orbital symmetry for this intermediate, right? Then, because we would experience some problems into the synthesis of those DNs, and also because we were curious about if the possibility to expand this chemistry for other more extended silane nucleophiles, we were thinking about these two possibilities to expand this 5-5 sigmatropic paradigm. One of them you can see is somehow a propyl silane that has been combined with an acrylate and is we can see that as an extended propyl silane derivative, the first transformation that I show you. And on the other way, if you just reverse the order, the location, let's say, of the triple bond, then you could be seeing this as an extended version of the aryl silane, E9s, both of the cases. Both has been prepared, but new chemistry as well developed to prepare those substrates with palladium chemistry. So then, when we tried the propargylated standard substrate, we were a little bit disappointed, I can say, right? Because turned out that this is nothing else in our hands. And I didn't say anything, but this work, by the way, is with Jorge Martin, one of our talented PhD students, Wei Chens and Catherine Golob. But let's keep going, and I will show you a picture of our little Jorge soon. So then, this is the propyl silane extended, and then we saw that actually the reaction took place in a highly selective manner in the ortho position. It was very efficient reaction, by the way. But then in our mind, it was like, well, we did all this effort just to do one more of the sample of the propargylation reaction. Still a nice molecule, but it was not our first idea. Then we went actually for the other inverse E9 nucleophile. And it turned out that this one, it was able to involve the whole conjugated system. And then we got a very clean reaction and very efficient, by the way, to create the new carbon-carbon bond into the paraselectivity, and probably the involved intermediate could be seen as a vinyl alanine iodonium in this case, right? So it seems like and something that is not easy for us to understand still, that the triple bond is playing a highly important role into the stereo control of these reactions. Then the pi intermediate that I was telling you that is supposed to be, and this is just a nice curiosity about the stability of these pi intermediates that are being formed after the initial decelerative transmetallation, right? So we were, by DFT calculation, we were kind of curious about what it was the conformational, rotational, to do a rotational scan around the carbon-iodine-carbon bond, right? To see what is actually the minimum of energy for this pi system, this pi stack system. And then, as you can see here, actually the pi stacking is quite of marked. And then the minimum, there's a sharp minimum into the energy in the rotational conformation, and when the two fragments are located above 9 degrees or so, right? Then we wanted to just to show you some molecules, right? To show you some more molecules of this type. And this is work with Jorge, Martin, and Wei. Then these are some of the pairs where as you can see here, if you choose one or the other enine, you can access in a very efficient and reuse-selective way to either the 3,3 compound or the 5,5 corresponding derivative. Here you have another example, in this case, bearing a bromo-deficient electron withdrawing group or pseudo-electron withdrawing group into the iodine, thiophene. We can also do this reactivity using different kind of deficient fragments into the enine, not only esters or phosphonates. It can also be, you can also use a simple pyridine ring. And this is a very recent result and very interesting one, right? We never had in the CUH alylation chemistry an heterocycle in this position, but now we are seeing this in a very efficient manner. Ketone groups, amides, alkylated secondary amides, and so on. I wanted to tell you about a couple of examples that are especially curious here. So for example, maybe somebody in the audience is already thinking about this, but if you block the possibility for either the standard propergyl silane or the standard allyl silane of the natural position for the CH functionalization, in other words, you install, for example, a two methyl groups into this iodine, and you expose this iodine towards the CH propargylation reaction. The reaction is indeed keeping working, it takes place, and the regioselectivity is fully derived to the method position. And something related is happening in the case that you're using the extended allyl silane version. And then again, if you block the para position, then you are deriving the reaction towards the method CH functionalization, also in a very regioselective way. So then let me just give you a possible rationale that we are managing in the group when we think about those substrates, and I'm using for this the paratholil iodine, right? So then what we do think that is happening is that the reaction indeed is taking place at the para position, and then once that you have this hybridized carbon, sp3 hybridized carbon in this Wheeland intermediate, you are opening the possibility to do a migration into of one of the two groups, and turned out that apparently, based on the result, the enine fragment has a higher ability for the migration in this reaction. This is just one of the possible explanations that we are having here, right? So then, so far, I have shown you that we were able to work with this 5,5 sigmatropic rearrangement, just considering the dienes, enines, and then if you take a look at the distance between the iodine and the para position where the CH functionalization is taking place, you can see that the distance is more or less matching the distance between the substitution with the electron withdrawing group, the place where the sigma bond is forming with the iodine, the place where the transmetallation somehow is taking place, right? The formation of the alanine iodonium intermediate, and the final of the final carbon involved into the reactivity. There is quite of a match in the distance, right? So then that is an obvious extension of this reactivity could be to consider another silane nucleophiles like benzyl silane, where the distance between the para position and the benzylic carbon in the silane will be approximately more or less the same, right? So then for this, we choose two different substrates. We go one of them much more simpler, the simple benzyl silane, or even a benzyl silane that has an additional metalloid, and there is mostly boron or tin in this benzylic position, a substrate that open an interesting reactivity as well. So then just for the sake of time, I don't want to take too much time. I just showing you here some of the samples of this reaction that, by the way, has been developed in parallel by the group of Professor Ivan Hayats in Adelphi University in New York and our group almost simultaneously. This is just an example of selected CH, para-CH coupling with the benzyl silanes. Here is a nice comparison depending on the allyl silane of benzyl silane over the same one iodonaphthalene. You can regioselectively obtain the two compounds. And Then when it comes for the, and this is just for the sake of time, I'm not taking too much more time, but I wanted to show you that in these particular cases, in the benzyl silane, when you incorporate another metalloid into the benzylic position, you can open a question, of course, because both of the metalloids could be transmetalate into the iodine center. Then when you expose those gem dimethyl or these dinucleophiles to the iodine upon the activation, actually what turned out to happen is that the transmetallation still is a decylillative transmetallation. In other words, silicon is leaving leaving the scenario faster than the boron, and then you probably can imagine, and this is what our DFT calculation suggests, that the formation of this boryl quinoid iodonium intermediate that upon the possible transition state of the sigmatropic rearrangement will give you a compound that it could be C as a CH allylation, as a CH aryl borylation of the iodoarine, and it can also be depicted as a metal-free IYAMA coupling of this carbon silicon bond, right? This is just some of the samples that we have been prepared with this methodology, nice boronate compounds. And then interestingly, in the cases, for example, in the case of where we are using thiophene derivative iodines, in the cases that the silicon group gives a little bit poor reactivity, we switch in the preparation of the nucleophile, the silicon fragment by, for example, tributyltene fragment, and then seems like the tene reagent is transmetallating faster into the activated iodine and more efficiently for depending of the substrate. So then, so far I have told you 3,3, property relation, allylation, 5,5, stienyl, bis-enanyl, enanyl and benzyl silanes, different types. Then, of course, the expansion is tentative, right? Expansion of this reactivity towards a larger iodines and a larger conjugated system is where we were wondering recently. And then we end up trying to name this the ultra remote iodine guided CH coupling. One needs appealing names for ourselves, right? To motivate and to name new reactivities. So for this, we just engineering two new 7-carbon 6-pi electron substrates, which are these two that I'm showing you in the slide. One of them will be an extension of the benzyl silane with a triple bond in this position, propiolate group in the para position of the benzyl silane, and the inverse substrate, which has a more related propergyl silane nature extended by the presence of this aromatic ring, right? So maybe you can already wonder what happens when we try the reaction with the extended propergyl silane. This is a beautiful work done by Jorge Martin. So then we again once more end up having the selective CH orthono in this case one functionalization of these two iodonaphthalene. In other words, the pseudo-ortho functionalization once more, or in other words, not involving probably into the reactivity the aromatic ester, right? However, I wouldn't be here saying anything about this if it would be because it was not working. We were happy to verify that actually when you invert the location of the triple bond, and this is something that we learn on the basis of the reaction of the beautiful reactions with the enines, then when you invert the location of the triple bond, the possibility to engage the whole system, it is open up, and then you are ending having a remote CX coupling of this standard benzyl silane, in this case in the carbon 6 of these two iodonaphthalene. This compound has been fully confirmed by the X-ray diffraction. And this is my almost the last slide. Again, in this reactivity, you can imagine, and then this is what they say, our DFT calculation, kind of well stabilized by pi stack interaction that is maybe accounting for the nice regioselectivity. And if you decompose, if you do a formal decomposition of the two possible fragments of this pi intermediate, reducing the naphthalene, you will have the 2-iodo-naphthalene, and that will be the almost orbital surface of this 2-iodo-naphthalene, and that it could be seen as a cationic standard conjugated LUMO. And then you can see here that there is a nice and accurate orbital matching, and in what it could be seen as a 4-point interaction pi stacked pair. So then this is the conclusion slide. We have been learning a lot with this chemistry. So main group chemistry, and this is why the name of the title, it was telling us many things to the group for some years. We love iodine, we love silicon, we love also boron as well, as you were seeing. Then that's why we end up having this name of the group, BZ-bond, right? The boron, silicon, and iodine, and these are the family of nucleophiles that we have been applying into this CH reactivity. And then, of course, this is I would like to acknowledge the people who do the chemistry, who bring the ideas, who give us, Alice and I, all the motivation there. Darwinians, fantastic, young and motivated people, fantastic collaborators, and fantastic and necessary, as Pera was saying, foundation, financiation, and funding to keep doing some chemistry. Thank you, all of you, for your kind attention.
And a super talk. Wow, the hypervalent iodine chemistry has been extended quite a lot by all of these fantastic results. Well done, super stuff. So again,
thank you.
All now open for questions from the audience or the panelists. Don't take down your slides yet, just in case we've got to go back to them.
Yeah. Okay. I tried. Sorry. Yeah, you're
okay.
right. Yeah,
I thought you were just showing me the new car you bought last week.
Sorry.
Yeah, no,
Time to move on, perhaps, Pat, because we are quite behind schedule, because it's taken longer to Anna with her presentation. Yeah? It's your job. Sorry.
Okay.
No, Anton is fine. Totally. Thank you.
Yeah. Okay. Well, thanks again, Anna. Super stuff, and lots of interesting chemistry from hypervalent iodine chemistry. Okay, Anton, over to you to introduce the last speaker.
Last speaker is Professor Ana Pla Quintana. She comes from the Institute of Computational Chemistry and Catalysis from the north, from the University of Girona. So, Ana, thanks for accepting the invitation, and go ahead. Audience is yours, yeah?
Can you please confirm me that you can see my...
Yes, perfect. See and hear.
And does it move?
Yes.
Yes.
Okay, perfect. Thank you. Okay, so let me first start by thanking the organizers of this meeting for giving me the opportunity of being here today. Indeed, it is an honor to be able to explain you our endeavors towards the development of reactions that capitalize on the rich reactivity of vinyl metal carbenes that we develop at the University of Girona. So why center our attention on vinyl metal carbenes? Because these are a fascinating subclass of carbenes, because they can also be viewed as an hybrid between a carbene and a metal-stabilized allylic carbonyl. These species are very good in cycloaddition reaction, but furthermore, they show electrophilicity at the carbenic, but also at the remote or vinylic site. Due to the highly intrinsic reactivity of the carbenic site, selective reactions on the vinylogous one are very challenging. Pioneer work on that was carried out by Davis back in the 90s by developing this reaction of this rhodium vinyl carbene with the cyclopentadiene. But let's center our attention on how metal vinyl carbene can be accessed. The conceptually more straightforward strategy is by the composition of a diazo derivative or a tosyl hydrazine or hydrazone derivative. Indeed, steryl diazoacetates or enol diazoacetates have been decomposed under rhodium catalysis to generate rhodium vinyl carbines by the groups of Davis and Dole. More recently, the group of Xu had shown that under silver catalysis, tryptosyl hydrozones can also be decomposed very easily to generate silver vinyl carbines. But there are also more elaborated strategies. For instance, Barren developed the decarbonation of 1,3,5-cycloheptatrienes that very efficiently generates rhodium vinyl carbines, or the group of Cossy developed the opening of cyclopropenes through gold catalysis. And also, I want to highlight this work by the group of Fürstner, in which an unorthodox hydrogenation of an alkyne, in which the two hydrogens are getting attached to the same carbon, also generates the ruthenium vinyl carbine. But this example by Fürstner is not the only example of methodology that is able to transform an alkyne into a vinyl carbine. Indeed, the carbene-alkyne metathesis is an excellent option to do this job. This reaction consists of the reaction of a metal carbene and an alkyne, and can generate this metal vinyl carbine. Indeed, first studies were carried out by Dodds that indeed developed the so-called Dodds reaction, in which a chromium Fischer carbene reacts with diphenylacetylene to generate naphthol derivatives through the intermediacy of this chromium vinyl carbine. An interesting aspect here is that if the reaction takes place in an intermolecular setup, this gives a very nice entry to polycyclic structures. And indeed, when we started the project I'm telling to you about 10 years ago now, there was this very nice example by the May group in Houston, in which this diazo derivative was intermolecularly reacted with this alkyne to generate this rhodium vinyl carbene that then reacted on the carbenic side through a carbon sp3 hydrogen insertion to generate these very nice polycyclic scaffolds. The reaction showed very nice diastereoselectivity, but there was no example, and here it was not possible to develop enantioselective versions of this process. So we asked ourselves if it was possible to develop a stereoselective carbene-alkyne metathesis cascade. At this moment, we were working quite intensively with rhodium-1 binary derivative catalysis. And so what we developed was a reaction in which we had a tosyl hydrazone, an alkyne, and an alkene. This was our substrate. And under the influence of rhodium code BF4 and BINAP, under hydrogenation, we generated catalytic species that indeed decompose the tosyl hydrazone to generate the carbene. And from here, the carbene-alkyne metathesis takes place. And then it was possible to carry out an enantioselective cyclopropanation. Interestingly, this process takes place without the need of a base, and was carried out with very high enantioselectivity. At this point, we decided to see what happened if we located an allene in the final position. And here we unlock a new and for us very interesting reactivity that allows us to generate these four methylene tetrahydropyran derivatives. Indeed, we studied the mechanism of this transformation by DFT calculations, and I here summarize a bit the mechanism that takes place through the formation of the rhodium vinyl carbene through the carbene-alkyne metathesis. And then we have a reaction of this rhodium carbene with the external double bond of the allene that generates initially a strained rhodocyclobutene that is not stable but reorganizes to this intermediate that very much resembles the structure of a trimethylene methane. And from here we have an inverse beta hydride elimination that locates this hydrogen to this position here. And in this rhodium allyl intermediate, the sulfonate that came from the decomposition of the tosyl hydrazone coordinates to the rhodium and gets inserted selectively into the position. As a continuation of this work, we also wanted to study what happened with the substrate featuring two alkynes in its structure. And here we were very happy to see that the carbene-alkyne metathesis take place, not just one, but twice, and generate this rhodium vinyl carbene that even has more extended unsaturation, but to which the sulfinate in this case reacted not into the carbenic site, but on the vinylogous one. That's very interesting to us, and we studied a bit more this process. Competitive experiments showed that the attack was intermolecular, and we were also able to show that indeed the proportion of attack at the vinylogous or at the carbenic site depended on the electronics on the sulfinate, and as these were more electron withdrawing, we increased the quantity of attack on this carbenic site. That shows us that we should be able to functionalize the vinylogous position of these in situ generated vinyl carbenes. And I guess that you agree with me that the sulfinate attack is not a very canonical reaction of carbene. So we wanted to go to, let's say, more classical carbene reactivity. So we checked the literature, and indeed there were some precedents on selective vinylogous reactivity. And for instance, it was shown that it is possible to carry out an NH insertion into this vinylogous position. Also, a carbon sp2 hydrogen insertion of an indole. And more recently, also a carbon sp2 hydrogen insertion of anilines in a process that, in this case, has been carried out stereoselectively. But a point that I want you to center your attention and that really caught our interest was that in quite some of these reactions, rhodium and silver gave complementary reactivity. For instance, in this OH insertion reaction, when rhodium is applied as catalyst, we have almost selective reaction on the carbenic side. Whereas if we use silver, the reaction takes place with very high selectivity on the vinylogous side. That happens with this OH insertion, and there was an example also on NH insertion. So with all of these precedents, we sent as an objective to develop carbon sp3 hydrogen insertion into the vinylogous position. In order to tackle this objective, we thought that we needed a silver catalyst, and so we contacted our friends in Huelva, Dr. Ana Caballero and Professor Pedro Perez, that have worked very intensively with the silver TP catalysts that are very, very efficient in the insertion into carbon sp3 hydrogen bonds, being able to insert carbenes even into methane. So we thought that we had the excellent catalyst to do the job, and then we needed to have a good platform. And for that, we found in the literature a very nice example from the group of Doyle and in collaboration with Sue, in which they show that in such kind of platforms, the carbene-alkyne metathesis under rhodium was efficient. And then in their hands, by controlling the electronics and also the sterics of the rhodium paddle wall catalyst, they were able to have carbon sp2 hydrogen insertion either into the primary carbon sp2 bond to generate this kind of compounds or the secondary one. So we started this project with several challenges. First, the silver catalyzed carbene-alkyne metathesis had just been described in one example with the simple silver salt. So we didn't know if the silver TP catalyst would be good catalyst to promote this transformation. We could also have the competing carbon sp2 hydride insertion in this position once the vinyl carbene is formed. And more important, we need to control the insertion either onto the carbenic or onto the vinylogous side. In any case, we went to the lab, and with this substrate, we set out the reaction, and we found the formation mainly of two compounds, two diastereoisomers indeed, that result from the carbene-alkyne metathesis. So we were happy to see that silver was a good catalyst to promote this transformation, and then insertion into the secondary carbon sp3 hydrogen bond. But we were very happy to see that we had the formation of a very small amount of a compound in which the primary carbon sp3 hydrogen bond was inserted into the vinyl logos position. In order to disfavor the formation of these compounds, we thought of removing this area here so that this position was no longer benzylic and so was less activated. With this new substrate, we optimized a bit the conditions, and we were very happy to see that we were able to optimize with quite high selectivity the formation of this 7-membered ring that comes from the vinyl logos attack on the sp3 hydrogen. We studied the scope of this reaction. The reaction is quite general. We can locate different substituents in this aromatic ring here that comes from the phenylacetic acid. The best selectivity is obtained with the unsubstituted substrate, but we can have more electron donating and electron withdrawing groups. It is also true that with the electron withdrawing ones, the selectivity is a bit compromised. We can also introduce substituents in the dimethylamino benzyl group, and here we have very nice selectivities, regardless of the electronic of these substituents being introduced. So at this point, we were very interested to see what was the mechanism operating in this reaction. We started by synthesizing deuterated analog with deuteriums in this dimethylamino position here. And what we observed was that the deuterium was very selectively inserted into the carbenic site on both compounds with no scrambling into other positions. So with this information in our hands, we started the DFT study. And indeed, in the literature, there were different mechanisms proposed for this carbene-alkyne metathesis. And since this had not been studied with silver, we evaluate all of them in our system. So we evaluate the cyclopropanation of the alkyne, also the 2+2 retro 2+2 strategy, the 5-endodic cyclization, and also the attack of the diatom onto the silver-activated alkene. And from all of these pathways, the one that was more efficient was the formation, after the formation of the silver carbene, was indeed a 5-endodic cyclization attack that indeed was barrierless, followed by a 1,3 isomerization of the silver to generate this silver vinyl carbene. Indeed, here, the barrier to overcome was the formation of the carbene that is a bit less than 12 kcal per minute. From this point, then, we wanted to have more information on how this CH insertion takes place. Indeed, we thought that initially there was a high rate shift, and from here we can have either an Fe2 mechanism that gives rise to the functionalization on the carbenic side, or an Se2 prime mechanism that indeed explains the formation of the attack on the remote or vinyl logos. And indeed, what we find out with the calculations was that the attack on the carbenic site is the thermodynamic product of this transformation, whereas the formation of the vinyl logos compound is the thermodynamic product of the transformation, as it has a slightly lower barrier of formation. The small difference between the two transition states also explains why it's difficult to have higher selectivities, and sometimes we have mixtures of the two compounds. More support on this pathway was obtained by evaluating the difference in energy of these barriers with the compounds with the different substituents. And we were very happy to see that with DFT calculations, we were able to predict the trend that we had observed experimentally. At this point, we were very convinced that the in situ generated metal carbines were very nice and powerful intermediates, and we wanted to center our attention to another type of compounds, which are nitrile halides. Nitrile halides can be generated, for instance, by reaction of a nitrile with a metal carbine, and that initially generates a metal bone nitrile halide that can decoordinate the metal to generate a metal and bone analog. These kind of compounds are excellent 1,3 dipoles, but when in these substituents here we have groups that with nucleophilic behavior, we can also have 1,5 electrocyclization, and there are multiple examples of formations of 5-member heterocycles through this strategy. But if we extend a bit more the conjugated system here, 1,7 electrocyclization is also possible. Indeed, this 1,7 electrocyclization gives rise to these 7-member azepine derivatives that generally also undergo 1,5 hydride shift to recover the aromaticity on this part here. This strategy, it is described, but just in a very few examples. So Padua, back in 1975, in a paper in which he describes a lot of different reactivity, gives an example of a ring opening of an aziridine that generates this halide that suffers this 1,7 electrocyclization. Sharp also described in 1992 that the 1,3 dehydrochlorination of this kind of substrate also gives rise to nitrile halide that also undergoes this reactivity. And more recently, the group of Krasavin reported this reaction from these kind of diazo derivatives. So with this precedence, we thought that with the platform that we have, we should be able to develop CAM17 electrocyclization one-time shift cascade process. Indeed, here there were also multiple challenges to be faced. First, the nitrile could react to the initially generated carbene and or to the vinyl carbene that's generated upon CAM. Also, there are almost no reports on the intermolecular trapping of vinyl carbenes, and here we needed this intermediate to react intermolecularly and not suffer, for instance, the carbon sp2 hydride insertion. And finally, we need to control the 1,5
Anna, I think we okay. I think you might have just paused there, Anna.
Sorry?
I think you paused. So maybe if you could say, maybe go back about 1 minute. Your screen paused.
Okay, so you cannot see my screen moving?
No, I can. You know, it's just that you talking paused as well is what I meant. Only in the last 10-15 seconds, that's all.
Okay, now you can hear me?
Yes, yeah.
Can I continue then?
Yes, please.
Okay, thank you. Okay, so here we had problems. So we started the reaction and we observed that we always had a mixture of 2 products, the desired one, but we had this also the formation of this compound that results from the product which has had a carbene-alkyne metathesis, but then undergoes the carbon sp2 insertion into this bond. And so the nitrile halide does not compete efficiently with this transformation. Luckily here, we had a substrate in the lab that had nitrile already embedded in the structure. And we thought that it would be a good idea to try it just to see if we could have a more extended cascade. And the first reactions were a bit difficult, but upon some optimization in which the temperature and the concentration were key, we were able to find out conditions in which we formed this very nice compound here, in which 5 new bonds and 4 new rings have been formed in just one reaction step. And furthermore, the reaction was completely diastereoselective. So we studied a bit the scope of the reaction. The reaction is quite general. We can have here in these aromatic rings electron withdrawing and electron donating groups, with electron donating groups giving better yields. We can also introduce substituents in the benzonitrile ring here, and in this case, it's better when we have electron donated one in methyl to the nitrile. And not unexpectedly, when we have substituent and methyl in this position here, we obtain a mixture of 2 regioisomers. Here again, we were very interested to see how was the mechanism of this very complex transformation. And as in the previous case, we started by deuterating the substrate. And what we found out upon the reaction of this substrate was that there was no scrambling and that the deuterium was incorporated in this position next to the isoindole ring. Also, if we map a bit in the substrate the bonds that are being formed in the transformation, we can see that we have a formal nitrile trifunctionalization that's quite bizarre, let's say, transformation in the literature. So we again rely on DFT to unravel the mechanism of this transformation. And I show you here our results. So we start by formation of the rhodium carbene. That's a process that takes some 14 kcal per mole as a barrier. And once we have this rhodium carbene, then the carbene-alkane metathesis takes place. That similarly to the silver project takes place through a 5-endodic cyclization, followed by the rhodium isomerization to generate the rhodium vinyl carbene. At this point, we have a second molecule of substrate attacking the rhodium carbene. And so we generate this compound, so the nitrile halide, from which the rhodium decoordinates. And here we have the 1,7 electrocyclization that generates the 7-membered ring. From here, we could have the 1,5 hydrate shift that would recover the aromaticity here, but quite surprisingly, this process was quite high in energy. And we found that indeed it was more favorable that the rhodium generate the second carbene, so the carbene in the second substrate. And we had the second carbene-alkyne metathesis with barriers that were, all of them, very affordable. And in this compound, indeed, the newly formed vinyl carbene is very close in proximity to this nitrogen. And so there's a nucleophilic attack at this position that generates the isoindole ring. And in this substrate, we do have the 1,5 hydrate shift that recovers the aromaticity on this ring here. And finally, to explain the transformation that we observe, there's just a ring contraction left that we postulate that takes place by the catalysis of a proton and upon workup of the reaction. Okay, so that's the end of the talk. Just as a summary and outlook, I hope that I've been able to convince you that the CAM cascades are very, very powerful to access intermediate vinyl carbene metal complexes that are otherwise inaccessible, and that they have very exotic reactivity. We've been able to show the reactivity with rhodium-1, with the silver, with interception of the vinyl ghost position, and also this rhodium paddle well extended cascade. And just to finish, I want to thank all the people that have been involved in the work. Professor Anna Roglans, that's co-leading with me the experimental results, and that has been always a great support. Professor Miquel Solan, Dr. Albert Puete, that have supervised the DFT calculations, the collaboration of Dr. Ana Caballero and Professor Pedro Perez from the University of Huelva, and the three main players of this project. So Oscar, that was in charge of the all initial results with rhodium-1, and Alex and Roger, that worked very intensively to have the results on the silver and the rhodium paddle well. Also, thanks to the financial support of the Ministerio and the Generalitat de Catalunya, again, to the organizers and all of you for your very kind attention.
Anna, thank you very much. Superb finish to a superb day. Again, it's open for questions from the audience, either participants, attendees. You can add your question into the Q&A. You can raise your hand. So, Anna, for sure, the reactivity of metal carbines never ceases to amaze me. You know, you can almost get it to do so many things that just about you can never predict in some ways. And I suppose you've done excellent work in terms of the rhodium complexes and various cascades, and more recently, I suppose, silver. Do any other metals get involved in metal carbines that do useful chemistry, or are they the main two? It's not my field, so it's maybe a very ignorant question, but I'm just asking nevertheless.
Yeah. I mean, there are quite a lot of different metals that are very useful to do reactions with carbines. Is it true that with rhodium, the degrees of selectivities that have already been achieved are superb, and they work with very low catalyst loadings, but there are quite a lot of groups looking for other metals. So silver is a very good option. There are quite a lot of work also with copper that also gives very nice results. And then I would say that a lot of different metals. Iron is also being studied in some ways. So going to, let's say, more first row metals, it's a trend that it's quite often followed in this field.
Yeah, I suppose I know Anita McGuire's working in Cork in Ireland here. She does a lot of copper carbene chemistry. So there's another metal. We've two questions coming through. One from Arian. You go first, please.
Yes, thank you very much. Anna, very nice chemistry, obviously. I was just wondering, so here you focus on rhodium or silver, let's say, separately. What about cluster compounds where you have both metals maybe being involved, or maybe even heterogeneous systems where, I don't know, I mean, if you think about nanoparticles, there are ways to design catalysts, you know, with different metals. Could that maybe add to, let's say, a third option to use carbene chemistry and to, I don't know, diversify the organic structures that you can make with those catalysts?
I don't know if I understood you correctly. You are saying heterogeneous catalyst?
No, well, not necessarily. I mean, you could have cluster compounds where rhodium and silver are, let's say, both incorporated. Or if you want to put it more general, you could say hetero bimetallic or hetero multimetallic systems. Would there be a way for those metals to induce cooperativity in the sense that you could maybe design, let's say, new reactivity patterns apart from the ones that you have discovered and described?
Yeah, okay, now I get you. Yeah, I think using different catalysts may also be a good option to combine the different properties of the different metals. But also maybe more than that, what I think that it would be very interesting is to combine, for instance, metal catalysis and organocatalysis here, because in quite some cases, so especially in these very extended cascades, the problem, for instance, we've been struggling quite a lot in the last part to have stereoselective results. And with the results that we have by DFT, we have the feeling that what happens is that generally in the key moment, the metal is no longer very close in proximity to where the action takes place. So maybe, and for instance, in the last case, we had this final proton ring contraction that if that could be assisted by an organocatalyst that was nearby or that was part of the catalyst somehow, that maybe could give the answer to access these kind of compounds in an anti-selective manner. So I think that you are right that combining different things in close proximity so maybe can help to have better or new selectivity, let's say, in these kind of compounds in which you have quite a lot of different options that can take place. So thanks for the question.
Great. Thanks, Arian. And final question then from attendees. Xavi, again, you're up for a question. If you can please ask it. Hello, Xavi. Hand is raised. Nobody home. Okay, disabled talking. We'll stop then. So maybe, and if you can take your slide down, we'll finish up.
Yes, sure. Thanks.
So can I, on behalf of the Division of Organic Chemistry in EuChemS as president, and thank again Anton as the local organizer for this event. And this is would you believe it, this is now the 12th society/country that we've highlighted across the last number of years, 4 per annum. So, you know, I gave Anton a template. I have no role whatsoever in choosing speakers, but I'd like to congratulate him on his choice of speakers. We've had a very interesting morning. We heard a lot about the Catalan Society in terms of the history, you know, founded in 1931. Some background material, just in case. 184 people registered for today's event. We started off with about 70-ish participants, give or take. We peaked at about 102. And then with lunch, we went back down again. So it's a sort of a typical day, I suppose. But, you know, 4 hours is a long time, so to speak. But that's a really, when you look at virtual conferences, you know, across the world, you know, getting about 100 people to attend virtually, I would rate as a success. So well done for all the local work in trying to get people to register. A lot of people registered in Dublin as well, by the way. So we're happy with that. So it was interesting to hear about the 12 universities, 5 of whom have chemistry programs, about the research centres. I would have been vaguely aware of some of those, having visited it, and all the events that the Catalan Society organises, the journals, the awards. And, you know, it's great to see. And today is really a snapshot of current successes in organic chemistry across the region. So it was really super to hear about the work from Daniel, who started off with clip-off chemistry, which is almost the antithesis of click chemistry. So you're actually using something to get something back off again. So that was an interesting concept, useful for supramolecular chemistry, for MOFs and COFs and whatever else you're having. And really simple olefin reactions of ozonolysis and all that sort of stuff that we would all heard about as undergraduates. But I don't think people would have thought about it to do what he does in the way he develops those supramolecular chemistry. So really fantastic work. Then we had Arian talked in one of his 3 areas, the advanced stereoselective synthesis piece, and it was all about decarboxylative alkylations to begin with, starting with palladium, then went to nickel. And then we had a bit of cobalt and photoredox catalysis. So, you know, finishing up then with some tetrasubstituted olefins, that making them stereo-defined, which is really a big problem in organic chemistry. And he's able to link that then to known drugs. And then finishing up with some interesting work of using the radicals to ring open the 1,1-propellane to make those bicyclopentanes. As you say, the very interesting bioisosteres that lots of people, including ourselves, are actually working on just now. So that was interesting. Then we went over to the world of carbohydrate chemistry, glycochemistry, glycans, protein glycosylation, understanding the mechanisms. This is from Carme, and the N-acetylglucosamine transferase was one of her enzymes that she looked at, and there was a really fantastic work in terms of quantum mechanics, simulation, protein structure, mechanism, the development of transition state mimics, therapeutic agents. That was a fantastic talk. Then we had Perry talking about, again, some more, if you want to call it, bio work in engineering aldolases, increasing the tolerance of the enzyme to new nucleophiles, new substrates, many FSA variants, and more recent work then on sort of artificial metalloenzymes, either using palladium for boron and heptide couplings, but also azomichael addition of amines. So really protein engineering at its finest there, really interesting results. Then over to Oscar, who told us new tricks for an old reaction, asymmetric hydrogenation. And really that was a superb example of the use of oxazoline-based ligands, nitrogen to phosphorus bound, and how that makes or bonded, and how that makes a huge difference in terms of the excellent levels of enantioselectivity. But again, a common feature across all of the talks today that was underpinned by a very deep understanding of reaction mechanisms, quadrants, molecular mechanics, DFT studies, all that sort of stuff. So well done, Oscar. And then we finished up with two Annas, so I better define which Anna I'm talking about first. So Anna from Barcelona in the University of Rama and Lul first. So all about teaching hypervalent iodine new tricks as well. So, well away from the oxidizing agent and maybe just the dull alpha arylation stuff that these reagents did first, but really outstanding orthoallylation, parallylation, and I hadn't realized that a 7,7 sigma tropic urea reagent was even possible, let alone the way she got it to go. So that was really interesting stuff. And we finished up then with Anna, again, this time from Girona, just to differentiate vinyl metal carbines, again, rhodium metalysis, silver metalysis, silver mediated carbines, the divergent chemistry you get from that sort of changes. Again, very nice Angevandikami paper, the 2023 on the silver carbines, and then finishing up with nitrile ilids. And yeah, again, a deep understanding of reaction mechanism. So it's back to Aryan's quote at the end of his talk. I'm trying to remember. It's not about knowing, it's about understanding. I don't know if I'm paraphrasing it, but it's something like that. And that was a key feature, I'd say, throughout all of the 7 talks. The deep understanding you have of the work that you're doing, and also the elegance in the way you presented the work. So it's an outstanding addition to the series of highlighting organic chemistry across Europe. And for your information, for 2025, we'll start with Switzerland in Q1, then we'll go to Greece, Serbia, and Lithuania. And after starting this about 3 years ago, where it was, I would say, a bit of a challenge to get people to go first and all that sort of stuff, now they're beating me over the head to try and get on the list. So we have 2025 already in place, and the 4 for 2026 as well. So watch this space for information about the Swiss event, which will be, I think, in February. I'm not quite sure which Friday we'll go with yet. But again, on behalf of the division, thanks to Anton, and very sincerely, thank you to all of the fantastic 7 presenters, and thanks to all the attendees, and I hope you enjoy the symposium. So enjoy the rest of Friday, and surprising that none of you mentioned that Barcelona Football Club are 9 points clear in the La Liga. I'll just put that in there as an interested observer. I'm sure you're all happy about that. So have a great rest of Friday and a good weekend. And again, thanks. And I'll pass it over now to Anton to say some final words on behalf of the local host and organizer.
I don't have much to say. I just would like to echo, Pat, your comments in extending our thanks to you all, participants and speakers, for engagement and support and participation. So that's all, and it was our pleasure organizing or helping organize this event. Nothing else. Thanks to all. Thanks, Pat, for trusting us.
No, you're very welcome. And it's great to see so many friends I've met before, and I hope to see many of the rest of you again, should I either visit Catalonia or you ever visit Dublin. So that's an open invitation. Okay, so thanks again. I'm going to pause the recording now. Bye-bye.
Gracias a todos.